సో మీరు డిస్క్ ఇచ్చేటప్పుడు ఇది పెట్టుకోండి ఆన్లైన్లోకి రికార్డ్కి.
యా, ఇది ఆఫ్ చేసేది ఎట్లా?
మీరు ఆన్లైన్ పెట్టండి, ఆఫ్ ఏం అవసరం లేదు. ఇప్పుడు కాదు, ఆన్లైన్లో చెప్పేటప్పుడు పెట్టండి, అక్కడ ఆన్ చేస్తారు. ఈ టిప్స్ రెండు ఓపెన్ చేసి పెట్టండి.
Let me see how it goes.
Just leave 4 chairs. This side two, that side two. You can occupy. You can do a little back. Leave 2 chairs this side, 2 chairs that side. Remaining. Cheapest.
एक मिनट, आपसे कोई इंट्रोडक्शन दे दें उसको, उस टाइम पे भी लगा लीजिए। ये क्या ऑनलाइन मीटिंग की
Okay, so anyway, the informal formal inauguration will start tomorrow. So I'm not going to talk more on. Originally we thought we'll have orientation in the beginning. Today 9:30 to 10:30 because there are some delay in trains, your train and other people. So I will just quickly myself, Raju Gopal, the professor in the Department of Plant Science, who is going to be a main organizer from University of Hyderabad, and Professor Echiro Takahashi from Okayama University, who is a real expert in chloroplast biogenesis and engineering. So he is going to be a foreign faculty. It will be another 10 days. He will be with us. About him, you can see the posters. He is a well-known molecular plant biology and molecular biologist. Worked on a lot of chloroplast biogenesis, especially in chlamydomonas. And for entire his career, he spent on the biogenesis of a photosystem 1. You have a photosystem 2 and photosystem 1. He spent his entire time or no on photosystem 1 biogenesis, and where they have generated a lot of mutants and a lot of side directed mutagenesis, and to understand the how the assembly of various proteins in photosystem 1. So with this, I think all of you, I request you to interact with him on another 10 days and get knowledge from photosynthesis related molecular biology tools. And myself, Raju Gopal, we also work on a lot of photosynthetic aspects and abiotic stress, which will be discussed later. Maybe later I will introduce a little bit more. So tomorrow in formal meeting, inauguration, we'll discuss more about it. And Professor Raghavendra is here. He is the most senior person in the department who is going to deliver. I think now I request Namrata, you can introduce Raghavendra sir. And afternoon post lunch, we will know each other. We will sit together and we will know each other. Ice breaking session will be there in the afternoon. For the moment, let's start the program, and we'll have a ice break session around 2 o'clock. Okay. With this, Namrata, you can introduce. And I just want to say that I would like to thank Professor Echiro Takahashi coming all the way and spending with us almost like 2 weeks, taking a lot of pain. And his wife also is here. You can interact with her. Professor Misan, we will say that Echiro is the first name and Takahashi is the last name. So in Japan, what do you say that Takahashi sensei? Sensei means like a teacher, sensei, right? Yes. Yeah, sensei.
You don't need to say sensei.
Yeah, but yeah. So it's like a respect. Kozumi-san, she's one accompanying him, so you can also interact both of them.
Good morning, everyone. It's my pleasure to introduce our distinguished guest today, Professor A.S. Raghavendra, the UoH Chair Professor at the School of Life Sciences, University of Hyderabad. Professor Raghavendra is an internationally acclaimed plant physiologist with over 4 decades of pioneering research work in plant molecular physiology and biochemistry. His research focuses on understanding photosynthesis regulation, stomatal signaling, plant stress physiology, and the interplay between chloroplast and mitochondria. He is renowned for his seminal and research work on nitric oxide signaling and reactive oxygen species, which has transformed our knowledge of how plants respond to environmental stress, and also has helped designing new approaches to enhance plant resilience and productivity. He has done his PhD from Sri Venkateswara University, Tirupati, and has worked in Germany as a Humboldt Fellow. Professor Raghavendra is a recipient of J.C. Bose National Fellowship, Government of India, and is a Fellow of INSA, NASI and NAS. He has also received prestigious recognitions including the INSA Young Scientist Award, Academy of Sciences Young Scientist Award, UGC Career Award and the INSA Professor T.S. Sadasivan Award. With over 250 influential publications, contributions to landmark books and reviews, and services on editorial boards of leading scientific journals, Professor Raghavendra continues to shape the field of plant science and inspire future generations of researchers. Let us all welcome him with a huge round of applause. Thank you.
Well, thank you very much. First of all, let me thank, I will stand as long as I can, then sit down. First of all, let me thank Professor Rajagopal and Professor Takahashi-san for having arranged this nice meeting, very useful for youngsters and also for us to share some of the thoughts. So today I am going to talk a little bit about actually photosynthetic evolution in eukaryotes. You will not find a very focused kind of approach. I will tell a few points which are interesting, definitely, even for non-plant science people. And whatever I could get from the literature, I try to present here. No, maybe you can use that. Well, as all of you know, our Earth has a long history. So what you see here on the left, oops, oh no, it doesn't work. Can you change this to laser pointer?
Yes.
Mobile, can you just change it? So what you see here is millions of years ago, because we are somewhere here, and I am talking of 500 million years ago. What about this? Yeah. Yeah. So life has originated long ago with mostly say like bacteria, and then later in terms of photosynthesis, a group of plants which are called as charophytes. So they are mostly something like between algae and bryophytes, as you can see, chara and related species. So they're all green algae, and from them we have these bryophytes which have originated. Then we have pteridophytes, and later on gymnosperms, and finally angiosperms. As you can see, angiosperms are very, very recent. They have evolved only about 150 years ago. And if you see this Mesozoic and Cenozoic, it has also a lot of how do you call yeah, this I think I will go back later. Now, just to be brief about photosynthetic oxygen evolution. Oh, sorry, I should use that. Yeah, these photosynthetic organisms have evolved with a very interesting kind of background. Oh, okay. Yeah, that may help this also. So we have actually an anaerobic eukaryote which has engulfed an aerobic bacterium. See, aerobic bacterium can make ATP by using oxygen, whereas this anaerobic eukaryote can give protection to this. So this has what you call as eaten and kept the aerobic bacterium inside, and then started what you call as evolving. So you can see, as it engulfed, it also developed chlorophyll and ability to make ATP, and so this starts with actually mitochondria. So when you engulf a mitochondrial cell, it represents the primary symbiosis, and later when a cell has already has this mitochondria, it now engulfs another eukaryote containing chlorophyll, then it becomes what you call as a complete photosynthetic organism. So this development of mitochondria followed by, yeah, this is much more clear. As you see on the top, the host cell will engulf a bacterium which is simple enough, and this bacterium slowly starts developing the kind of inward projections, and which becomes something like a very similar to mitochondria what we know of. So, at this stage, this particular microbe with the bacterium inside now engulfs chloroplast. So, if you see with the time, so we have an ancestral prokaryote which is simple, not much differentiation, but then you have this infolding of plasma membrane which helps in developing the nucleus. So, we have a nuclear envelope, enclosing nucleus, and development of endoplasmic reticulum, and at this stage it engulfs an aerobic heterotrophic prokaryote, and so this is something like a first stage of symbiosis. So, we can say primary symbiosis. Then we have a secondary stage where you get the chloroplast to be engulfed. So, this prokaryote with mitochondria and chloroplast can be now engulfed by another prokaryote. So, this becomes something like a tertiary symbiosis, which I will show you later on. So, we have so stepwise incorporation of organelles, mitochondria like structures, chloroplast like structures, and ultimately getting into a stage where we have the present day what you call as bacterium. So, that is what is represented here. This is all endosymbiosis considered when an aerobic bacterium engulfs an aerobic bacterium, leading to a symbiotic relationship that benefits both. When I say benefit, the aerobic bacterium can make ATP using oxygen, and this is equivalent to mitochondria, and but then this oxygen is not good for the other one. So, it is toxic to the anaerobes. So, this anaerobe provides the ingested nutrients and protection to the engulfed aerobic bacterium. Yeah. Now, if you see this on a time scale, as I have already mentioned, we have talked of several billion years ago, 3.5 billion years ago, we have a very simple prokaryotes. So, they are all supposed to be represented by the kingdom Monera. They have this very old archibacteria, mostly methane producing, salt loving and heat loving bacteria. Then we have the evolution of typically cyanobacteria. I think you all know that cyanobacteria are the oldest kind of algal systems, very what you call as resistant to harsh conditions in the environment. At that time, of course, then organisms like E. coli, Mycoplasma, and so on. And as the time progressed, 2.5 billion years ago, these cells have started engulfing mitochondria and then chloroplast. So, both of them, they become part of eukaryotic cells by this endosymbiotic theory, primary and secondary, and at a later stage it becomes a tertiary symbiosis as well. So, by about 1.5 billion years ago, and even at this stage, we have several of them, eukaryotes which are small, falling into the category of protista, and then we have fungi, plantae, and animals, and so on. And as this evolution progressed, we have some interesting things being developed. So, viruses have developed, and these viruses, since they are capable of infecting several things, some of them are actually what you call as pathogenic, and they do not have a proper DNA, sometimes RNA, never both. So, these are actually quite pathogenic, and from here we have even simpler kind of organisms which have only RNA, and later on these also have evolved into something more interesting. Proteins are also pathogenic. You must have heard the name of prion and causing bad cow disease. So, these are proteins. So, we have a whole cell containing nucleus and then organelles. Then we have a cell having either DNA or RNA, and then we have cells which are having only RNA, and then we have organisms which are made of only proteins. So, this is a quite interesting kind of evolution going on. And if we go back to the Earth cycle, as we mentioned, we are talking of Mesozoic era, which started more than 250 million years ago to about 66 million years ago. So, this is very old and this is later. So, we are talking of minus million years ago. So, during this time we have Triassic period, Jurassic period, Cretaceous period. I think this Jurassic period, many of you might be familiar with the movie. I think you have seen Jurassic period is very well known for actually the age of reptiles, and these reptiles, what you see in the film is a depiction of dinosaurs, but it doesn't mean only dinosaurs. There are a lot of other organisms as well. But then at the same time, the other side of the story is that there are also a lot of gymnosperms coming up during this time. That's why I think the movie, they tried to depict a lot of these gymnosperms during that time. So when you see the movie, you have to see the science of it. That's more interesting than that. So during this time, we have gymnosperms which are dominant, and dinosaurs which were dominant, and later the flowering plants have started appearing. So during Cretaceous period, instead of gymnosperms, flowering plants became more and more. And at the same time, what you see down here is the earth itself. As all of you know, at one time the earth was all one piece of land, and later it started splitting. I think you know about this Gondwana split, quite famous in the history. It breaks down and produces, India on one hand, Africa on the other, South America on the other, and what you see now here is something like this. And then if we go into the recent times, so we are talking of the age at this time. So first of all, it was all animals, so mammalia were dominating. Later, horses and primates have evolved, and then in the so this is called age of mammals. So instead of dinosaurs, we have actually mammals. And during this time, we have also the organisms growing, and from small and like gymnosperms, we have now a lot of land plants evolving, and then there is an evolution of these. Many of these mammals started eating grass and started grazing. So this is quite interesting. Okay, so this is one part of the story. I have been mentioning about the earth and the life. But when you come to the evolution of photosynthesis actually is quite interesting. We can consider it at the organism level, and we can also consider it at the reaction center level, as all of you know. Typically, what we learn at this time about photosynthesis, I think you will hear more and more of it. We have sometimes photosystem 1, sometimes photosystem 2, both of them in higher plants. But there are organisms where you may have only photosystem 1 or only photosystem 2. So Professor Robert Blankenship is actually one of the authorities on photosynthetic evolution, and he has been trying to describe how the photosynthesis has evolved based on the evidence. And what he said is the earliest organisms were anoxygenic, and that the photosynthetic reaction centers were from a single source. So from this anoxygenic photosynthesis, reaction centers have evolved to become oxygenic, and then they become diversified. So when they become oxygenic, they also developed antenna systems, and then followed by carbon fixation pathways. So reaction centers are the first things to evolve, later diversification of antenna systems, and then the carbon fixation pathways. So this is actually a kind of evolution. Archaea is a very old one, where you have all the what you call is the type 1. I think this in here this is easier. Okay, this is quite clear. In olden times, we had only purple bacteria, and these purple bacteria were not evolving oxygen, whereas oxygenic phototrophs have developed later, and many of these were using two types of photosystems and two types of reaction centers. This story you all know. Reaction center, photosystem 2 has P680, reaction center photosystem 2 has P700. And please remember, maximum they can absorb is about 700, never more than this. So we are talking of only red, not far red. But when you look at the earliest organisms, now you can see here this bacteriochlorophyll, which we are talking of in case of purple bacteria, it has only two types of reaction center. It doesn't evolve oxygen, but it needs very high light. So, something like far red light. I will say this kind of red requiring bacteria are known only in purple bacteria, and a lot of interest on that. And if you look at this connection, as all of you know, photosystem 2 and photosystem 1, they are connected by a cytochrome B6F complex. So, this cytochrome B6F complex has evolved from a different form here. So, if you see here cytochrome B6F complex, it was only BC1 complex. So, this also has evolved, not only reaction centers, but also this connecting cytochrome complex. And at the same time, I should also mention that this type 2 or this type 2 kind of reaction center is based mostly on quinone. I think you must have already heard. Now, we talk of plastoquinone here. So, here also it is a quinone. So, it is a quinone based reaction center or photosystem. Whereas, when you come to photosystem 1, it becomes an iron sulfur type reaction center. So, a lot of details here. And at the other end of the story, you have again a red absorbing kind of reaction center, type 1 type of reaction center, which has iron sulfur type alone. Now, as all of you know, we can have either a non-cyclic electron transport, electrons moving from P680 to P700 and ultimately pyridoxine, whereas you can have only a cyclic type of electron transport with electrons moving from reaction center to pyridoxine and then coming back. So, we have again BC1 complex. So, the evolution has occurred in types of reaction centers, the type of cytochrome B6 or BC1 complex, and then how this electron transport occurs. Yeah, this is what is indicated here, and it is a simple way of representing the earliest reaction center is a very simple one. From here we have the evolution of type 1 RCs, mostly looking like photosystem 1. So, based on the PSA-A, PSA-B, or we have type 2 reaction centers where you have all this typically D1, D2 and then LM type of things. So, what we have is something like dimers, but heterodimers, whereas in between there are also reaction centers which are quite different from either of these two, and these are also of very high importance in terms of knowledge and in terms of what we call as understanding the evolution of oxygen. In fact, again Professor Blankenship has explained a lot. So, if you want to really know more, you can try to refer this, and in fact, he has written a detailed book on this, but this article is quite good, and as you can see here. Well, let me again mention how the evolution of organelles have happened. I think I have already mentioned with the time the ancestral prokaryote developed membranes, developed nucleus, nuclear membranes, endoplasmic reticulum, then engulfed mitochondria containing or mitochondrial type of the bacterium, and later engulfed something like, it all starts with unicellular algae, and when it engulfs unicellular algae, we have a prokaryote which starts looking like almost like a eukaryote with mitochondria and chloroplast, and then this whole thing sometimes it is engulfed by another one. So, we have a first stage of symbiosis, second stage of symbiosis, and then the third stage of symbiosis. Yeah, so this terminology is quite interesting. People try to compare it with the French words. There is a term called as pas de deux, step of 2, like what you find in ballet, and what people mean that this evolution of what you call as secondary symbiosis goes very well synchronized. So, it is no less than a ballet or maybe even more complicated. The tertiary symbiosis, which you find in many of them, this is even more complex. So, it is called Menai-Troyo and a blend of 3. So, this is a wine with 3 types of wines. So, they are quite different, but they evolve and work quite well. So, this is what is represented here. So, primary symbiosis occurred about 1.5 billion years ago. A free living prosynthetic cyanobacterium was engulfed by a heterotrophic eukaryote which has already an organelle inside, and this now takes in a photosynthetic kind of plastid and now achieves this secondary endosymbiosis. And this one can happen with either green alga or red alga. Both of them are used depending on the situation, but then we talked more about tertiary symbiosis, which is quite interesting. So these events, what we call of primary symbiosis, so where you have the chloroplast getting engulfed, and then secondary symbiosis where this is one organism, this is another organism. So as you can see here, both are actually motile organisms, and then they engulf the whole thing. For example, this organism is engulfed by another one, so it becomes tertiary symbiosis. And we have situations of where we have several other instances of tertiary. So let me just read this. Original symbiotic event is believed to have been a primitive flagellate protist and a cyanobacterium. And when it enters, there is a lot of shuffling. Some genes are kept, some genes are lost, and then it results in obligate intracellular chloroplast. Then in secondary symbiosis, we have a heterotrophic straminophile, again motile, and again there is a lot of massive gene transfer and loss of a few components, and then divergence into chromophytes. So chromophytes are all colored organisms. It may be green, or it may be red, or it can be brown, and so on. Then we have the secondary tertiary symbiosis, where a whole dinoflagellate is engulfed and tries to keep up this function of photosynthesis. And people believe that this kind of tertiary symbiosis is still going on. So I will tell you a few examples where we can see this in progress. So what you see here is something like Elysia chlorotica. I think you will be able to understand more. So Elysia chlorotica is a sea slug. It's an animal. It's like a snail, and actually it's quite interesting in several ways. What it does is it eats the brown algal filaments. Vacheria litoria is a typical brown alga, which is what you call a siphonaceous. So it has kind of continuous body. So it tries to eat this and sucks this chloroplast. So what is described during this process is that Elysia chlorotica is a sea slug which sucks or which steals chloroplast from the other host. So we have some details of this coming up. So the chromophytic macroalga Vacheria litoria undergoes symbiosis with E. chlorotica as a result of suctorial feeding. So this sucking itself is quite interesting, and this has been noticed in sea slugs, which are mostly they are all in sea. So then what happens is the nuclei are lost, only chloroplasts are retained, and sometimes these chloroplasts they all get what you call as deposited on the skin, and then this is now quite interesting. As you can see here, this is Elysia chlorotica, which has taken up a lot of chloroplasts, and you can see it keeps this green color, which also means that it is capable of using light and producing certain carbohydrate. And now you can see there are instances in say like our old literature that there are people who don't need any food. They can simply stand in sun and make a menace, and then they can have their own food making. So very similar and quite fascinating kind of organism. And so this doesn't happen from the birth. So this Elysia chlorotica starts acquiring this chloroplast when it starts eating on Vacheria. Then we have another example. It's called spotted salamander. As you can see, it is an amphibian, actually quite interesting, because this amphibian can lose parts of its body but still can what you call as survive. I think you all know about lizards. Many of the lizards, they can lose their tail, but nothing happens, but then this they go to a further level that they can survive. So then here we have what we call as Lotharella globosa, and this is again another interesting kind. Say Paulinella chromatophora is actually eukaryote, which engulfs another type of bacterium. I will come to it later, something like Cercocystis or sometimes Chlorococcum. It engulfs and keeps it during its life cycle. And so we have at least 3 examples which I try to tell in detail, but what you see here in this picture is also diatoms. As you know, diatoms are one of the oldest kind of organisms known to be capable of hosting a lot of smaller algal cells, and they are known to be what we call as symbiotic. And then there is a lot of discussion whether diatoms should be classified into animals or should be classified into plants. So as a plant scientist, we prefer to call them as plants, but then some people claim it as also animals. So now you see here, we try to see a few more details about sea slug, and it will be interesting to see this. ఎందుకు ఇది రావడం లేదా? Oops, sorry. No, it's not working there. రావాలి ఇది. ఇది ప్రెస్ చేస్తే రావాలి కదా. Yeah, this is what happens sometimes. అంటే ఇది పాయింటర్లోకి వెళ్ళిపోయింది. Yeah, now it will come. Yeah, now you can see this is the sea slug we are talking of, Elysia. It's it's trying to feed on this Wacheria, is a filamentous algae, although it looks green, but it's actually brown algae. You can see happily feeds on the algal filaments, and then these algal filaments, they are taken in, and all these chloroplasts, they go and settle finally on the skin. So it is described that it steals chloroplast from the alga and uses it for its own benefit, and for this its mouth parts are specially made for that purpose, to suck the chloroplast from the algal strands. So this goes on for a while, and as you can see, and these chloroplasts are incorporated, and they usually find their way onto the skin. So it is believed that these chloroplasts on the surface of the skin, they are significant enough to produce carbohydrates. And this topic is actually a lot of people debate and question whether they can really do this. Some people believe that these chloroplasts are not really sufficient to support the life of this marine slug, Elysia chlorotica. But what is known for sure is that when these plants, they go into something like a hibernation, they can go and survive with minimal kind of eating. So they may be producing some carbohydrate, not to the level of full kind of activity, but capable of allowing survival of the sea slugs. So these sea slugs have been the topic of study for several people, as you can see. Excellent examples for symbiosis. So we have an association of a mollusk and then algae, and then trying to what you call as function. So quite a few of them. Green sea slugs have been objects of curiosity since a long time, as you can see, more than almost like 3 centuries. And then there is a lot of variation. Each of this so-called I have mentioned only one example, but there are other examples. So all of them, they belong to a group called as Ascoglossa, and each of them has its own kind of choice. So not every species likes the same species, so they have their own choice of food like us, and then chloroplast functionality can also vary, but mostly they need something like a siphonaceous type of algae. And then it has also been found that most of these slugs, they prefer to have a brown brown type of algae rather than the green algae. And now when we have this tertiary symbiosis, what you can call them? Is it an animal or is it an alga? Well, I think it is an animal hosting an alga, so it's not an alga hosting an animal. So, but isn't it definitely a good example of tertiary symbiosis? So, this is what I was trying to mention. When the Elysia is born, we can see these eggs, they don't have any chlorophyll, they don't have any chloroplast, they are all plastid free. But as they grow, 21 days also, something like 3 weeks after hatch, they start learning how to feed. So, when they come in contact with Vaucheria, they start what you call as sucking the chloroplast. So, they change their shape, and then after say like a day after feeding, they are at that time they can use this material for their growth, and then this phenomenon is called as kleptoplasty. So, plastids are taken inside and kept closed in the animal. So, that is why they are all called as kleptoplasty, and about 5 days after feeding you can see a nice change in the color. The animal has become green because of this kleptoplasts, chloroplasts which are contained, and the adult one becomes more and more interesting. And as you can see here, we have a lot of plastids. It has grown and quite green, and they are not just green, you can see chlorophyll fluorescing, and if you look at under fluorescence microscope, they can be seen easily. So, they have this chlorophyll fluorescence, and now this is more a lot of more details. There has been a lot of interest in this plant, and it has become possible when scientists have developed a procedure to grow them. As you know, in many of the instances, if you are working with an organism, the moment you are able to grow them in the lab, the research goes on very well. So, that is what you see here. So, you can grow them in aquaria with sea water mimicking solution, but you need to also keep the temperature quite below. So, they have to be maintained about 10 degrees and dim kind of lighting, and you can grow and do a lot of work with this. And this is all examples that chloroplasts are functional. You can put light and then see the evolution. So, this is evolution of oxygen from the Vaucheria, which is a brown alga. You can see a good kind of oxygen evolution system, but Elysia also show this. It responds to light and dark, and then 25%, 100% light, and so on. So, the point is that the Elysia chlorotica, which is an animal and which has engulfed and sucked the plastids, it is capable of performing typically the photosynthetic activity. Yeah, the same thing. The cells are not green. Then, as they start feeding, they what you call as develop green color, and then in this case the alga is slightly different. This is Acetabularia acetabulum. This is also a siphonaceous type of production. Oh, well, actually this figure has a lot of details, and I think we will try to avoid it. Yeah. Now, a few more examples of the sea organisms which are quite interesting. So, this is another type of sea slug, Saccoproteus species, and this Saccoproteus species looks quite similar to an alga which is called as Chloropa, and you can see this is sea grapes. Okay, Saccoproteus, another species. This is actually is an alga. Chloropa is a kind of bryopsidal green alga from coastal regions, and then actually liked by people who are living in the coast, and that is why they are called sea grapes. Yeah, and I think I will consider this as another interesting example. We have what we call as spotted salamander, which is an amphibian, and this is the Latin name, Ambystoma maculatum. It incorporates green algae. What you have seen earlier is brown algae. It incorporates unicellular green algae, for example, Euphila amphilostomatis, and this alga is very closely related to Chlamydomonas. And let me emphasize that many of these tertiary symbiosis examples, an animal and alga, usually are very, very specific in their preferences. Yeah. So, this is salamander, although it can grow actually, and this is as I said, it is known for its ability to survive even if it loses limb and so on. And now you can see, for example, here this actually shows up a little greenness, although it is not very clear here. But when you look at the eggs, these eggs, they have this unicellular alga quite clear. So, spotted salamanders with single cell algal cells, Euphila amphilostomatis, and so it has observed that a lot of interest has been shown to study this, and they found that this chloroplasts, they get incorporated into the cell, and the mitochondria from salamander, they come and try to get around this chloroplast. So, then they have something like a very close association. The chloroplast makes carbohydrates, mitochondria, they also give the energy. So, all of them together, they respire using oxygen and provide carbohydrate and use up together. So, it is something like helping each other. Now, another example, Paulinella chromatophora, it is an amoeba, actually engulfing again unicellular kind of organisms. So, this is a scanning electron microscope, but if you look at under normal microscope, it appears like this, and it is a single cell, and inside you can see chromatophores. So, this has come from the algae, and these algae are actually either Synechococcus or Prochlorococcus. Prochlorococcus is one of the earliest kind of green alga. Synechococcus is earlier one. As you can see, it beautifully stays inside, and they both of them, they work together. So, unfortunately, it is a bit difficult to grow in lab, unlike the slugs, but then I think one group could grow them, and they have very clearly showed that this chromatophore is quite interesting, and you can analyze the DNA or DNA, and then you can demonstrate that it is of Synechococcus or Chlorococcus origin. Yeah, this is another example. A freshwater glaucophyte alga is incorporated into Cyanophora paradoxa. As you can see the name, it is quite interesting and quite a paradox to study this animal. And this is again an example of tertiary symbiont. So, this is the Cyanophora paradoxa. It tries to take in a cyanobacterium, tries to incorporate and can get on with photosynthesis by using both of them together. So, it's we can say it's a model organism where two cyanelles or two chloroplasts which are incorporated, and these chloroplasts are called cyanelles, and they are quite interesting. And this alga looks like it's very similar to a pathogen, chlamydia, to acquire plastids. So, Cyanophora paradoxa is compared with disease producing pathogen, chlamydia, but it can acquire plastid, and this next figure shows up how this can be done. I think you have already seen how when this mitochondria and chloroplasts, they are incorporated through stages: primary symbiosis, secondary symbiosis, tertiary symbiosis, and this tertiary symbiosis going on right now. And now this is an example of, we were talking of chlamydia, Cyanophora paradoxa. This Cyanophora paradoxa is an amoeba type organism. It can take in the what you call as bacteria and chloroplasts, and then this chlamydial partner, mostly very similar to this chlamydial pathogens, they are lost during the course of evolution. So, chlamydia are quite infective. So, during the process of evolution of these eukaryotes, they take up this chlamydia type cells, develop archiplastida, and then these are all plants or something like green kind of thing. But at the same time, chlamydia have evolved in other hosts, as you can see here. They are known to be quite pathogenic. They can infect a variety of organisms, humans, cattle, fishes, mammals, and you can see pigs, birds, even smaller organisms. So this is that's why they are called as quite paradoxical to see that chlamydia-like bacteria, they can donate to photosynthetic organisms on one hand, and they can continue their life and start and be pathogenic on the other. Yeah, to end up, let me comment about this tertiary endosymbiosis, which has been known in case of volvox. I think volvox you must have heard at several instances. This is a multicellular organism made of actually unicellular organisms. So it's something like a colony, and then inside this colony we have certain dinoflagellates, and then in these dinoflagellates, some of them are quite interesting. And a good example of this dinoflagellate is a species called as Carenia. Carenia is a dinoflagellate which is famous for producing massive red tides. For example, if you have a chance of going to Florida at a given time, particularly when the temperatures are high, it produces a massive red color. And this massive red color is because of the enrichment of dinoflagellate Carenia, but one has to be very careful. This is quite toxic. So one should be careful when swimming in these areas. And then why I am mentioning about this Carenia is that this can donate, or it can go into symbiosis and become a part of diatom. So this again another example which is quite interesting. I think it should be the last one. Thank you and for your attention.
So if you have any questions, you can discuss with sir.
Well, evolution is a what you call as fascinating topic. It's very difficult to understand. I don't work on this, just to be frank. I work on the interaction of chloroplast and mitochondria. That's a topic by itself. And I also work on signal transduction in stomatal guard cells.
Any questions?
Either they should have understood completely or they have not understood at all. Yeah, please. Allowed. Yeah. You have a mic? Yeah.
So in Elixir how we call it a symbiosis?
Why do we call?
It's a symbiosis.
Elysia?
Yeah.
Oh, I think you didn't see that earlier slide. Yeah. See, the egg is colorless. It doesn't have any alga. As it grows, when it comes in contact with Vaucheria, this is a siphonaceous type of alga. They are all continuous tubes. They are not individual cells, not like say like Chlamydomonas. And this can grow only when it comes in touch with this. And as it comes in touch, it starts developing mouth parts and starts what you call as taking up it sucks. And later you can see about 3 weeks after hatch and about 5 days after feeding. So this has taken in chloroplast. So it's basically an animal, but sucks plastids, retains them. And most of these plastids, they go and stay in the surface skin. That's why there is a lot of discussion whether they are all. So if we can have that capacity, if we can humans suck plastids from, how do you call, leaves and retain them. It's not that easy. When you eat, what happens? Everything is digested. So it's a kind of almost like a miracle. It has developed that specialization that chloroplast is not what you call as digested. It is pushed into the skin and retained, and it is retained in terms of function and structure, both. So you have seen that when you look at the, oh, not here, no, yeah, there is somewhere I have shown you the chlorophyll fluorescence. So chlorophyll fluoresces and they can evolve oxygen, and you can see it's almost like the algal cells, some of them. So they are functional plastids are retained.
But for algae,
For algae, well, algae can get a lot of material from the what you call as the animal. Also, algae is also protected from oxygen. Many of these photosynthetic organisms, they also need the proper environment. So some of them, they like this inner kind of atmosphere. Not that it's very ideal, not like they can grow outside also. So it is a kind of both, helping each other.
but the idea I got is they're extracting the plastics.
Extracting and keeping it
For the algae.
intact, plastid is kept.
Yeah, so for the algae it's harmful, right?
Not exactly. For example, if it is harmful, what about this function? We have we are looking at only chloroplast. The plastid only is evolving oxygen here. If there is already a harm made, how can you expect this activity to go on?
But plastics are
Active. So that means the life is maintained. Plastid life is maintained. If you consider an organelle, whether it is living or non-living, it is living. Yeah. So it must be getting some kind of benefit from the host.
Sir, one question. After the consumption of bacteria, filamentous alga, so does the does it need any other kind of source like food source?
Yeah, this is what I said. When you have this green kind of animal, slug, people started arguing, well, it can live, without eating its own food. If you leave it in the sun, it can survive. So a lot of calculations were made, that so many plastids are there on the surface. So these plastids are sufficient. Personally, I believe that amount of food the chloroplasts make is not enough. If you consider the skin surface area and the surface of plastids, you can calculate how much carbohydrate it can make. So that kind of carbohydrate is definitely not enough to make the slugs survive under normal activity. What happens is, as I said, they also have their own ways of what we call as requirement of food. So when the temperature is normal, they move around. They are actually slow moving, so they can try to detect some food around. But if the weather is not very good, it becomes either warm or too cold, they try to settle down on the sea surface and they try to go into something like, I will not say hibernation, because hibernation is given for bigger animals. So they try to stay in kind of resting kind of stage. So in kind of resting kind of stage, whatever light they get at the bottom, this chloroplast can back up. Minimum kind of food they can make. So the consensus is that this algal systems in the slug is sufficient to make them survive under kind of inactive period.
Yeah, there's one more question.
So
You can use my.
Yeah.
this organism is in marine ecosystem, right? So there would
No,
be
I think your mic is not working.
Present.
Yes,
Yeah, so I was saying this organism is in marine ecosystem. So there would be other brown algae as well. So why this one is, this sea slug is selective to this brown algae only? Is there a difference in composition of the plastid or both?
Well, that kind of thing is very difficult. For example, nature has evolved in such a way that organisms have likings and dislikings. Suppose I say you can eat every food, you will strongly disagree. When it has evolved, if you are in a different kind of environment, you will eat everything. When you have a, what you call as chance of saying no, you are saying no. So this specificity, I will try to compare with the similar system in symbiotic nitrogen fixation. In symbiotic nitrogen fixation, the legume has a very specific association with a specific microbe. Actually, microbe not only species, microbe is also species and cultivar stage. So it has such a high degree of specificity. So this liking and disliking has evolved.
Any more questions?
Sir, during the evolution, these plastids they are retained or every time we need to capture again in every life cycle? These plastids they are retained in the evolution in the ratio or
No. When this sea slug starts producing egg, they are not retained. Eggs are free of plastids.
life cycle
Yeah.
it has to
It has to again start eating Vachyria and acquire. Yeah. Okay, maybe.
Any more questions? Thank you so much, sir, for such an interesting and insightful talk on evolutionary aspects of photosynthesis. We truly appreciate your time and presence and the valuable thoughts you have shared with us. Thank you so much, sir.
Thank you all.
So the next speaker, Professor Eiichiro Takahashi. So I said, he's last almost 40 years, almost 4 decades, right? Almost 4 decades. He's working in photosynthesis related topics, and his research is very focused on algal photosynthesis, especially Chlamydomonas reinhardti. So, and also as I said, his lab is established with a lot of equipment with full of biochemistry and molecular biology instruments, where he may be focused. Last another you need to go 10 11 12 talks. In that he may cover from from biogenesis to the functional aspects, as well as the biochemical aspects, and also one talk will be also on structural aspects of photosystem 1. So I think tomorrow we'll be going to have more of formal introduction in front of Vice Chancellor. Again, I would like to thank Chiro for coming here and sharing your knowledge with our the participants, with the students. So please go ahead.
Microphone is on. You can hear me? Okay. Thank you very much. Very kind introduction, and I'm very glad and honored to be here again in Hyderabad. This is my I don't know, fifth visit. Since I first visit to Hyderabad, I became a very fan of Indian food, spicy food, and very very nice. And okay, so first I introduce myself. When I started my this research on the photosynthesis was just a bachelor's thesis. It was 79.
Yeah.
So at that time I was working with thermophilic cyanobacteria.
That's what I said, 4 decades.
4 decades, okay, yeah, more than longer than. And also, at that time, why I used thermophic cyanobacteria was the reason was the proteins of thermophic cyanobacteria is very stable. So you can isolate the proteins or protein complexes that's usually fragile. We can isolate and characterize these complexes or proteins more easily. But I felt that at that time this thermophic cyanobacteria was not amenable to molecular analysis like recombinant DNA or transformation, and at that time no genetic information was available. And then much after I got a PhD degree, I worked a little bit, a few years in the plants, the spinach. But later I decided to use the green algae, alga, the Chlamydomonas reinhardtii. I just started to use Chlamydomonas in 1989, and that time just as I did some the work in the University of Geneva in Switzerland to learn the molecular biology of Chlamydomonas, and then at that time just they developed the selectable marker for chloroplast transformation. So then I joined this project and wanted to know to knock out or modify the photosynthetic genes encoded by a chloroplast genes using Chlamydomonas. So that is my starting point of using the research on photosynthesis using Chlamydomonas, because genetic information and chloroplast and nuclear transformation, although I haven't experienced the mitochondrial transformation, that is nothing to do with photosynthesis, I think. So I did the nuclear and chloroplast transformation using Chlamydomonas. So that is a very powerful, this alga is very powerful organism as a model organism. And there are also many the mutants in, deficient in photosynthesis. So there are many resources we can use. So that's why I'm, how to change this? This is pointer?
You can change this.
No pointer.
You can change the slides.
Only change slide. Okay. So here I'm going to molecular mechanism of chloroplast biogenesis, in particularly the more introductory talk, and then in my other lectures I will going to give you later more specific topics I will talk. So of course we know that plants and algae contain the chloroplast, but this was so this is just a summary of the 3 genetic system in the Chlamydomonas reinhardtii to show the pointer is here. Okay. Let's sit down. Okay. Okay. So this is Chlamydomonas.
Excuse me, can I use it?
Pointer. This one? Okay. Of course, the nuclear genome has the large size, about 110 megabase pair. And the chloroplast genome has only 205 kilobase pair, much smaller. And mitochondrial genome in Chlamydomonas is even smaller, 15.8 kilobase pair. And genes encoded in this genome is 585, about 20,000 genes in nuclear genome. But in the Chloroplast genome contain only 99 genes, and mitochondria even smaller, 37. So this gene in the chloroplast and the mitochondria also contain some genes encoding RNA for use for gene expression, ribosome RNA gene, and also transfer RNA genes, and also in the case of chloroplast, it contain RNA polymerase or the translation, some of the ribosome RNA, and also some ribosome proteins. Not all. Some are encoded by nuclear gene. Some are encoded by chloroplast genes. So this is a kind of a minimal size. And chromosome and copy number, the nuclear genome has 17 chromosomes. This is haploid, okay, not diploid. And chloroplast genome, Chlamydomonas has one chloroplast. Unlike plant cells, plant cell has many, tens or more than 100 chloroplasts, but in Chlamydomonas it's only one. And there are many mitochondrial genome, but it's about 130 mitochondria. Each mitochondria has 130 copy number of mitochondrial DNA. So, and usually we grow the Chlamydomonas in haploid. So if you have a mutation in the nuclear genes, if it has a phenotype, you can observe the phenotype because it's a haploid. If the cell is diploid, one of the pair of the genes, if that is wild type, you don't see phenotype, right? So this is very easy to screen the mutant that is deficient in your specific phenotype of your interest. So inheritance is Mendelian nuclear gene in Mendelian law tetrad in the segregation. And in the chloroplast, mutation is inherited in maternal. Maternal, it's we distinguish this male or female, but it's designated as a mating type. This is mating type plus and mating type minus. Okay, the mating type plus is inherited from mating type plus cells if you genetically cross. But in the mitochondria, the inheritance was paternal, so from the mating type minus cells, its genetic information is transferred to the progeny. And if you introduce foreign genes from outside the cells, the gene DNA is integrated into the genome randomly, in the case of nuclear genome. But in the chloroplast genome and the mitochondrial genome, introduced DNA is integrated by homologous recombination. So you can, if you have homologous sequences in the introduced the DNA that are targeted to the right place of the DNA. So you can change specifically the genes of your interest in the case of the chloroplast and the mitochondria. That is very convenient. This one. Oh, no.
Keyboard. Keyboard.
Keyboard.
Yeah.
Here. Okay. So this is just summarize a history of the, not the whole history of the chloroplast, history of the discovery of chloroplast or characterization of chloroplast. I just took from some the source book or Chlamydomonas source book, the recently published in 2023. So it's described that in 1905, the plant chromatophores results from the uptake of cyanobacterium by eukaryotic ancestral organism. That is some prediction kind of things. The evidence was not shown at that time. And then 1962, these two scientists observed some fibris in the chloroplast using the similar green alga Chlamydomonas moybusi. So probably they claim that this DNA in the chloroplast. And 1963, Saga and Sega and Isida, they actually purified and characterized CP chloroplast DNA from Chlamydomonas cerevisiae. So they found that the Chlamydomonas had the chloroplast contain their own specific DNA. And then later, 1967 and 1970, the endosymbiotic theory was further established. So Professor this time very detailed history was presented. And the next is that how the chloroplast DNA looks like. So since 1979, the chloroplast genes were being sequenced. This means that the specific gene was cloned and then sequenced, not entire chloroplast DNA. And then from 1980 to 80, restriction maps of chloroplast DNA were constructed, okay, by digesting different restriction enzyme or combination of two enzymes. So then you can connect a fragment. And then it was found that the chloroplast DNA is circular. It's not linear. So this characteristic of DNA structure is similar to bacterial DNA. So that is consistent with the chloroplast is derived from the bacteria, cyanobacteria. And since 1979, the mutations in the chloroplast gene encoding a photosynthetic apparatus were found. So that clearly shows that some genes in the chloroplast DNA encode photosynthetic protein. Okay, and since 1973, chloroplast protein synthesis was being characterized. So one of the way to identify which proteins are encoded by chloroplast and which are encoded by nuclear genes. In the case of the chloroplast proteins, encoded proteins, if you add the antibiotics of the spexulmycin or something like that, so then this specifically blocks the ribosome, bacteria type ribosomes. So it blocks the synthesis of chloroplast encoded proteins. And for example, if you use cyclohexamide, that blocks the cis translation of the by the cytosolic the ribosome. So then you can assign which by using a labeling experiment. Okay, so So it becomes clear that some chloroplast proteins are encoded by chloroplastins, and the others, or majority of the chloroplast proteins are encoded by nuclear genes. So nuclear encoded proteins are synthesized in cytosol and transported, translocated into chloroplast somehow. And then 2002, the first complete sequence of circular chloroplast genome from Chlamydomonas cerevisiae was reported. And then, of course, there are so many, many errors in the beginning, but in 2018, very much refined chloroplast DNA sequence was determined. So the characteristics of this Chlamydomonas chloroplast DNA is GC content is low and AT content is high. But compared to the nuclear genes, the GC content is very high in Chlamydomonas. That is the main characteristic. So if you do a sequence of chloroplast DNA, there are so many A and T, A, T, 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 A, T, A, T, A. So it's somehow confuse us to determine the sequence. And also in the many years ago, the high GC content in nuclear gene is also difficult, makes more difficult to determine the nucleotide sequencing. So this is a genetic information. And 1988, Boynton et al. First reported a chloroplast transformation using a biolistic method. The biolistic means biological ballistic method is, maybe I will later in the other lectures, I will talk about how to do a chloroplast transformation, but here I just want to say small metal particle, for example, tungsten particle, it's like about, the diameter is about 1 micrometer. We coat the surface of the particle with transforming vector, okay, and then deliver this small particle into the cells, into chloroplast by particle gun. Do you know that particle gun? So we deliver this particle at a speed of sound, for example, 300 meters or 400 meters per second. So this is so small, so we have to work in a deliver must be work in the vacuum. Otherwise, the air they accelerate the particle speed and then go into. So it may kill many cells, but at a low probability, some successfully the particle is delivered into the chloroplast. So maybe later I will talk about how to select the transformant of chloroplast, because this is very essential, because as I told you, efficiency, or it's this ballistic method kills many cells, and also many cells are not targeted by particle. So relatively small number of cells are successfully transformed. So we have to select, for example, 20 or 30 transformants out of the millions of cells. So this is very important to selection of the transformant. But maybe we discuss later other talks about this. And in 1990, the chloroplast transformation was successful in tobacco in the plants. So this is more complicated process, but it's possible now. Tobacco is the method is very much established. And 1991, Goldschmidt Claremont in the University of Geneva reported the AADA cassette. That is confer the spextromycin, streptomycin resistant phenotype. So this AAD code for an enzyme that inactivate spextromycin. That is, spextromycin blocks the chloroplast translation. Bacterial type ribosome it blocks. No, sorry. Yeah, but if you inactivate spectromycin by ADA cassette expression, so then the cell can survive in the presence of antibiotic spectromycin. So only transformed the cells can survive, but non-transformed the cells dies. So this is very efficient selection method. Okay, so there are many types of the plastid. The chloroplast is one of the plastid. So echoplast, chromoplast, and chloroplast, leucoplast, amyloplast, elioplast, and proteinoplast. This is plant. But I'm working on the green alga. So green alga contain only, mainly, usually only chloroplast. And sometimes I will talk later, or sometimes other lectures, echoplast also generated in chlamydomonas. This is so others are not really found in chlamydomonas, but you can find in the plants. Okay. For example, amyloplast is a potato, store the starch. Chromoplast you can find in fruits, color. And leucoplast is no colorless, but it store something. This is a kind of leucoplast. No color, but it store other products in the cells. Okay. So this is the interconversion of the plastid. Okay, I skip. And this is a typical structure of the chloroplast. This is a chloroplast from spinach C3 plant. So this is a nice shape of chloroplast, and then as you know, the chloroplast is surrounded by 2 layers of membrane that is called envelopes, and outer envelope and inner envelope, 2 layers. And also in a membrane system, thyroid membranes are present, like here. This is starch. So this region is many layers. The thyroid membrane is stacked, so it's called grana G, grana stacks. So if you see this region, you can see many layers of the thyroid membranes. And inside this envelope of thyroid membrane is called luminal space. And this region outside the thyroid membrane is stroma that contain many enzymes, soluble enzymes. And this is also a chloroplast from grown in from the highlight condition, and this is the chloroplast from the spinach of low light grown. So if you look at the thyroid membranes inside the chloroplast, you will see the difference. Okay, the chloroplast from highlight grown the plants has less grana. Okay. the number of the stacking is lower. And this is a low light grown niche, and you can find easily the more stacks. So the reason is this is a strong light, so the light is not limited for the photosynthesis. So the plant has sufficient light. So chloroplasts do not need to capture light more. Okay, and in the low light grown cells, this need to collect more light. So the chlorophylls, photosynthetic pigments like the chlorophylls are present in the cytochrome membrane. So if you want to collect more light, you need more cytochrome membrane. So then to accommodate the large amount of membrane, you need to stack properly, right? If you have many clothes, if you just scatter on the floor, you have no space to live. But if you are nicely organized and in the furniture, you have more space. So that's the case. You need, if the chloroplasts need more cytochrome membrane, this is the best way to stack and organize. And this is a chloroplast from C4 plants, Z-maize, and mesophyll and bundle seed cells. So this contains PS1, PS2, this contains PS1, so that's also stacking structure is different. So chloroplast has different characteristics in the structure. And also if you look at the green algae, for example, this is a Chlamydomonas. So single cell and single chloroplast. No, the plant has many chloroplasts in single cells, but Chlamydomonas has only one chloroplast. So it's large, about 40% of the volume of the cell is occupied by single chloroplast. So it's a lot. So if you look at the shape, maybe it's difficult. This is a nuclear. This is a schematic. This is nuclear. So chloroplast is like this, single chloroplast. It's quite different from the plant. Okay, because there's a nuclear here. The chloroplast is like this is a section. So like a cup. If you cut the cup, you look like this U-shaped. So cup-shaped chloroplast is a characteristic of the Chlamydomonas. Okay, and the cytochrome membranes you can find here, and in the Chlamydomonas has less stacking, as you can see. Not a little bit stacking like here, but it's not like the spinach chloroplast. It's more like this, bundle seed cells of C4 plant. And this is a pyrenoid. It's contain the starch and also many enzymes, particularly Rubisco enzyme is concentrated here very much. But otherwise, the chloroplast is very similar. This Chlamydomonas chloroplast is similar to the chloroplast of the plants. So this Chlamydomonas is also can be used for model organism for studying chloroplast instead of the plants, because Chlamydomonas can grow very easily. You don't need much space. It's cheaper, right? And also, what is important is Chlamydomonas can grow without photosynthesis if you add proper carbon source like acetate. This is very important because you can isolate the chloroplast photosynthesis deficient, the mutant deficient in photosynthesis easily. Okay. Okay, if you look at the chloroplast DNA of Chlamydomonas, this is like that. It's very okay, circular DNA. Okay, and inner double-strand DNA. So inner circle is the transcribed this direction, okay, anti-clockwise, and outer chain is transcribed is clockwise direction. Okay, so these genes in a square is colored are the genes that is transcribed in a counter clock direction. Okay, and these genes are clockwise direction transcribed. Okay, and what is interesting is this is the genes classified. For example, this is green is photosystem 1 proteins, and dark green is photosystem 2, and blue is cytochrome B6F complex proteins, and yellow is ATP synthase, and this orange is Rubisco large subunit. Okay, these are related to photosynthesis, has function of photosynthesis. Okay, there are many genes, but for example, photosystem 1 forms a complex consisting of many subunits, and of course some very important subunits are encoded by chloroplast genes, but others, mostly peripheral subunits, are encoded by nuclear genes. So this is a case also in photosystem 2, cytochrome B6F complex, and ATP synthase. It's a kind of mosaic. Okay, chloroplast encoded subunit and nuclear encoded subunit are combined to form a functional complex. And Rubisco also large subunit, but small subunit are encoded by nuclear genes. And also the others, this violet is the brown related to expression of chloroplast genes. Okay, this is RNA polymerase, and this is ribosomal proteins, and okay, and also RNA genes, genes encoding RNA, tRNA and ribosomal RNA genes are present. Because all the RNA that is necessary for gene expression is encoded by chloroplast genes. Because RNA cannot be translocated from the cytosol to chloroplast. So all RNA must be synthesized inside chloroplast. Okay. And also other genes, these are, how to say, pink. It doesn't look pink, but some are openly framed that its function is not known, or FSH or YCF1, or these are other functions. Directly, indirectly, some are indirectly related to photosynthesis, but the others are not related to photosynthesis. And there's an intron. The gray is intron. For example, these genes, PSBA, is encoding one of the reaction center subunit of photosystem 2. This gene has a intron. So green part is exon. So this is the intron. Okay. And also other interesting gene is here. CHLN and CHLB gene. This is related to chlorophyll synthesis, protochlorophyll reductase. So maybe I talk later, but this is Chlamydomonas and some plants or for example, needle plants or moss plants can synthesize the chlorophylls in the dark, but many other plants, land plants, cannot synthesize chlorophylls in the dark. So there's a one step of chlorophyll synthesis pathway that is light dependent. But this light dependent pathway in the Chlamydomonas has the two types of the enzyme that is dependent on light and the other is independent of light. So Chlamydomonas, like as a moss or some pine needle plants, can synthesize chlorophylls if they, even when they grow in the dark. Okay, so this is important things. Okay, and also interesting characteristic is this circular chloroplast DNA has this region, this thick line is inverted repeat. So exactly the same nucleotide sequence, a pair of nucleotide sequence are present in the chloroplast DNA, but direction is invalid. So, okay, the sequence is exactly the same, but the direction is inverted. So that's we call the inverted repeats. And this is a single copy region, okay, here. So this is large, and this is smaller, usually. Which is larger, I don't know. It depends on the organism, it changes. But larger section, single copy region is, and also smaller single copy region, and also inverted repeats. That is the main structure of chloroplast DNA. But between this inverted repeat region, for example, this is large single copy region, and this is small single copy region, and this is inverted region, inverted repeats. Inverted repeats B, inverted repeats A, so direction is like this. But there's a flip-flop, the mechanism, so by a homologous recombination, this exchanges. So if that is the case, if it's exchanged, as you can see here, the direction of a short single copy region direction is changed, swapped. That happens. But it doesn't affect any expression of the chloroplast genes. So we don't need to care about, if we're working on the function of gene or working on the gene expression. We don't need to care about, but this happens, okay? And also what is important in the chloroplast has activity of the homologous recombination, as I told before. Okay, so then Chlamydomonas has single cell, a single chloroplast, and the plant has many chloroplasts. So in the case of plants, maybe there are many chloroplasts, so that you can, the cell can separate many chloroplasts, probably mostly equally. But in the case of Chlamydomonas, it has only single chloroplast. So when the cell divides, chloroplast must, division of the chloroplast must be synchronized with the division of the cells. And also a DNA of chloroplast DNA in the chloroplast is 80 copies, but must be separated. By chance, by chance, one of the chloroplasts, after the chloroplast is divided, one of the chloroplasts has no DNA and the others has all the DNA. If that happens, cell cannot survive without chloroplast DNA. So there's some mechanism how the chloroplast DNA is separated properly between both divided chloroplast. So this is about the experiment, how exactly the chloroplast DNA is separated. So before I explain this, chloroplast DNA is not present in the chloroplast, particularly the stroma region, as a naked DNA molecule. This is some the proteins bind associated with this chloroplast DNA to form a chloroplast nucleoid. It's not like a chromosome, but it's simple structure, but it's actually some proteins associated with chloroplast DNA. And then form some globule structure, maybe more than one chloroplast DNA nucleus associated to make a large forms. So this is a chloroplast nucleoid like this, globulous. And if you look at the confocal fluorescence microscopy, if you stain this, this is a yellow fluorescence proteins fused to one of the proteins that are associated with chloroplast DNA. You can assign by this fluorescence that where this chloroplast nucleoid are dispersed in the chloroplast. Okay, and then you can follow during a cell cycle, light cycle and dark cycle, G2 and SM and G0 cycle. So this grow the cell, and then the DNA is synthesized, and then its cell division occur. So you can follow the proteins of the associated with chloroplast DNA is by following. You can see how the chloroplast nucleoid looks like. So this large nucleoid particle becomes smaller, and then somehow dispersed to form like as if a network-like structure. Okay, and then cells and the chloroplasts are divided 2 times. So make 4 cells, daughter cells. So if this red is the the chlorophyll fluorescence. Okay, so it's, if you look at the fluorescence, the microscope, chlorophyll fluorescence is just coming much strongly. Okay, you cannot avoid this, but this shows that where the chloroplast is. Then it's divided into 4, you can see, and then this globular chloroplast nucleoid is dispersed, and then after division, it also again the globular structure appears. It grows and grows larger. This is the same thing, similar proteins, yellow fluorescence proteins. So this is how the 80 copies of the chloroplast DNA is divided into equally to after 2 division into 4 daughter cells. Okay. So what is important is this nucleosome doesn't move. If this protein, this chloroplast DNA is present in the stroma of the chloroplast, this should be moved, right? But it doesn't move, do not move. That means suggests that chloroplast nucleoid is probably associated with thyroid membranes. It's not free completely. That is the case. Okay. So another interesting feature of the chloroplast DNA is oh, sorry, this is something. When we do the mating type plus and mating type minus cells, the crossed, genetical cross, okay, then as I show in the second slide that chloroplast DNA or genetic information is inherited from mating type plus. It's not from mating type minus. So the chloroplast, this is not a cell division. This is a sexual cross. One of the chloroplast, the genetic information disappear. Okay, that is from the mating type minus, and one of the chloroplast information from the cell of the mating type plus only dominate in the after crossing. Okay, so this means that probably the after the fusion, the chloroplast DNA from the mating type minus should disappear or digested. Okay, that should happen, and actually this data show that that happened. Okay, this is a chlorophyll, and this is a DNA staining the overlay. So this is after crossing like this in the beginning, 60 minutes later, two cells fused. So this becomes from haploid to the diploid, and two nuclei is still present. And this is a chloroplast DNA. After crossing, still both from mating type minus and mating type plus chloroplast has the similar amount of the chloroplast DNA. Time is running out. Okay. So, but in 90 minutes later, no, the chloroplast DNA from mating type minus disappear, digested probably, right? Just happens. So this is just the same experiment. Okay. Okay, so if you look at here, so DNA is disappeared. This was present but disappeared. Okay. So this is a typical style of the chloroplast inheritance. I think I'm going to talk other lecture about a similar the subject. So maybe the later part is I will talk later in the later lectures. I'm going to talk about biogenesis more, but we have still time to do this later lecture. Okay. Okay. So I stop here, and I'm happy to have the question or from you.
So I would say, it is not that questions, if you have a kind of interaction, it is always better interaction. Any questions you can ask, like the main aim of this GIAN conference to interact with the students and inculcate whatever the subject, so you will be, kind of interested later. So that is the main note of this. So you can ask any kind of questions and interact with them. It is not like an exam. There is no exam here. So, yeah. If you have any clarifications you'd like to discuss? Please. Yes, press.
Sir, like you said that under the low light intensity, the size of the grana differs in this image. So does it affect the size of the chloroplast? Like if there will be more stacked grana, there will be larger size of chloroplast in both condition, or the size of the chloroplast will be equal?
You're talking about
Yeah.
this.
Very
So
good
in the
question. Yeah.
huh?
Very good question.
Sorry, the
Sir, will the size of the chloroplast vary
Ah,
according to the
size of chloroplast. That's I don't know exactly, but of course the color of the leaf changes, of course. Under low light condition, it's more stacked. The stacked region has, maybe we discussed other lectures. Major
The question is whether the size
size
of chlorophyll.
chlorophyll. Chlorophyll? The
Chloroplast.
amount of chlorophyll.
Chloroplast.
Chloroplast size is, I don't know exactly the size, because only the focus on the stacking. Size is, it depends on the organism, I think, but it's, I don't know, sorry. The size is maybe changeable, but there is no description of this aspect. Maybe that is a good point and to analyze, to check. You expect that if there's more stacks, the size of the chloroplast may increase. Maybe that but I don't know exact data or the evidence the difference in size, because people focus on the stacking. This is stacking. The number is drastically changes, so people just focus on this aspect. But the size of chloroplast is, I don't know if it's very important for the function of photosynthesis. Yeah. Other questions?
Question, sir. Here,
Yeah.
I have one query.
Your
As-
microphone.
Yeah, I think I'm audible. Yeah. You are telling about this chloroplast transformation. In that, particularly, Nt plus should be the one which is going to transfer to the next generation. So ideally, using Nt for chloroplast transformation, you should use always Nt plus strain. That is the ideal one.
Sorry.
For as you said, only the Nt plus strains is going to carry out the mutation to the next generation, right? In the chloroplast, whatever mutations you have made in Nt plus.
Mating type plus.
Yeah, mating type plus. So all the transformation will be done in mating type plus strain only. Or
Yeah, of course. That is basically yes. And after the chloroplast transformation, if we want to cross with some mutant that has, for example, the nuclear mutant, you can combine, chloroplast mutant and mutation and nuclear mutation together. But if you transform the chloroplast using a mating type minus, if you cross, you lose the phenotype. So of course, basically, almost all the case, we use a mating type for chloroplast transformation.
and added to this question, one more thing, Sensei, regarding the recombination. You said for transformation, the nuclear is random insertion.
Basically, recently we can tag
Specific homologous also. So in that case, if you specifically, I think the procedure seems to be almost similar. What do you feel? Like chloroplast transformation versus a nuclear transformation. If you use CRISPR in this case.
Sure.
So I think the procedures might be almost similar. Based on the recombination, I think the site might be.
Yeah, of course, it's the vector is different, the method is different. I have not experienced CRISPR-Cas9, but it takes more time than random insertion. So usually we use nuclear, the mutant as a host to transform of gene of your interest. Then the transformed the genes is randomly inserted in the nuclear genome, but it doesn't matter if it's expressed to complement the phenotype. So usually we use this method. But you need to screen because it's randomly inserted, so it may happen that second mutation may occur to if the transforming the vector inserted into some genes that may give you a new phenotype. But if that is not related to the growth of cell or photosynthesis, we don't care. But we need to isolate much more than one mutant transformant. And at least 3 transformants, we compare phenotype, and if they are the same, we can say that this is successful. And yeah, that's the case. So if you have the mutant, you can use this traditional nuclear transformation to complement, and also mutagenesis, cytodirected mutagenesis, or tag fusion. So if you have a nuclear mutant of genes of your interest, that's very powerful. But if you don't have, you need to use CRISPR-Cas9. Some people works very well, and some people don't do not. So that's sometimes risk. There is a risk of this method.
So we'll come back. I think we can have more discussion later. So 2 o'clock we'll assemble here. As I said, we'll have some ice break session. So we'll introduce ourselves. And the lunch is provided. There's just one more floor up. The steps are here. The guys will go guide you. So thank you, Vishnu, for
thank you. Thank you for your attention.
Information, and we'll come back around 2 o'clock. We'll have
sure
introduction to these students.
Sure.
Right.
Oh, yes.
say the GIAN poster?
The recent one I did or the
Poster. Oh yeah. Okay, so welcome back to the first day afternoon session. So today is more of, not really, it's more of introduction about the laboratory, the skills or tutorials. Is it okay? So what's your name? I forget. Your name?
Abhinek.
Abhinek. Okay. So as I said, myself, Raju Gopal, I'm professor here in the Department of Plant Science, and our laboratory, maybe now you can go through later the profile of lab. So we work mostly on photosynthesis aspects and abiotic stress related the proteins, the role of proteins or the organization of thylakoid membranes under various stress conditions. And we also been working on state transitions where photosystem 2 and photosystem 1 there is a balance. The excitation energy has to balance from photosystem 2 to photosystem 1. So that balance is called state transitions. Through light arresting complex, they migrate using a kinase called STT7 kinase, STN and STT7 in Chlamydomonas, STN7 in Arabidopsis and higher plants. So recently we also started CRISPR technology to editing some of the genes to understand the photoprotection mechanism, which is on thioredoxin and also autophagy related genes, and connecting again through more of molecular biology and physiology, biochemistry to understand again photoprotection is the main our goal, and also want to understand the lipid role in lipid accumulation in various stress conditions using CRISPR technology. These are some of the areas. Anyway, I am going to talk from tomorrow onwards about my research. So, as I said, I have about over almost 25 years of experience in this. I mean, if you say that from the my PhD days, it's almost like 30 years of experience on photosynthesis related aspects. Started with as I said, I also started cyanobacteria as a model organism, then worked on various algal and plant systems. So this is about me. As said, HRO we have already introduced. I think it may be introduced much better tomorrow in front of the Vice Chancellor, where he's, as I said, he have over 40 years of a research experience in photosynthesis, and mostly in plant biochemistry and physiology and molecular biology. The lab is mostly focused on molecular biology, where protein-protein interaction. Anyway, he is going to talk about it himself, so that it will be more interaction. So, one round of introduction yourself. So then, then we will go with the tutorial thing what we are going to do. Okay, so now, yeah, maybe we can start from this side. You can sit and tell, or you can stand so that everyone can see. You can just switch on that one, middle. So what are you doing? MSc or PhD?
Sir, I'm pursuing MSc in botany.
Oh, okay.
Good afternoon, everybody. My name is Sharmila. I'm from Odisha. Currently, I'm pursuing MSc Botany in Utkal University.
Myself, Rasulfidas, and currently I am pursuing MSc from Utkal University, Odisha.
My name is Satyajit Kujur. I am from Odisha, currently pursuing MSc Botany at Utkal University.
Myself, Dr. Madan Shankar Kalwa, Assistant Professor at Geetam University, Vizag. My major research area is mostly like photosynthesis and changing climate with respect to different types of food crops, most likely higher species.
And you'll be also one of the resource person. Right?
Good afternoon, everyone. Myself, Pranti Pradhan from Odisha, currently pursuing MSc in Botany in Utkal University.
Which college? Which university? Utkal University. All of you are from Utkal only.
My name is
My name is Asmita Kumar Desai. I am from Odisha and currently pursuing MSc in Utkal University.
Maybe can take.
My name is Kunjal. I am from ISET Tirupati. I am pursuing PhD and right now I am a visiting scholar at University of Hyderabad.
Yeah.
I am Nandana. I am just completing my Masters in Biotechnology from Pondicherry University.
Good afternoon, everyone. I am Vishal Jamni, PhD scholar from Goa University.
Good afternoon. My name is Tanuja and I am from Geetam Visakhapatnam, pursuing PhD under Dr. Madan Shekhar sir itself. So I am working on plant science and majorly pigeon peace response to abiotic stress and how it is tolerated.
Hello, I am Nalin Varghese. I am from Kerala. I graduated my MSc last year, and for the previous 1 year I have been trying for the CSIR net coaching.
myself, Shrija. I have recently pursued my MTech from JNU and now I'm attending this workshop to know more about Chloroplast Biogenesis.
Celia, you can start from.
Good afternoon all. I am pursuing my PhD. I am currently in 4th year. Good afternoon all. I am currently pursuing my 4th year of PhD in Professor Rajgopal Sir Lab. Currently, area of my work is under photosynthesis at drought condition in a plant and algae as a biofertilizer.
Good afternoon, everyone. I am Kanika Sharma. I have recently joined in PhD under the supervision of Rajgopal sir in HCL.
Good afternoon, everyone. I am working as a senior technical officer in University of Biotechnology. My area of work is isolation of actinobacteria from diverse environments, and we are isolation of secondary metabolites from actinobacteria and biological evolution. And apart from that, I would like to pursue my research work in the algal research. That's why I used to come this.
Right.
Good afternoon all. My name is Monica. I am pursuing second year PhD under the supervision of Dr. S. Venkatamohan sir. My area of work is especially in electroculture, focusing on a root rhizosphere with the influence of electricity in situ potential in molecular biology.
Myself, Ragini. Myself, Ragini. I am currently pursuing first year of my PhD under the guidance of Professor Rajgopal sir. Currently, I am working with the desert algae Cestria and studying some of the biophysical properties of that algae. Thank you.
stress and investigating the role in tackling.
Thank you. Good afternoon all. I'm Pavitra. I'm working with Rajagopal sir. I'm in my final year of PhD and I work with the modular organism Unariella salina. It's a photosystem remodeling in photocolouring.
I'm Shreya Sharma. I'm a third year PhD scholar under Professor Rajagopal, and my work entails studying the redox control in Chloroplast strain garden.
Good afternoon all. Myself Dr. Lakshmipati. I'm doing as a postdoc researcher with Rajagopal sir lab.
Good afternoon everyone. Myself Dr. Venkateshwar. I work as DST Inspect faculty in the Department of Plant Science in the University of Hyderabad with Professor Rajagopal sir.
Good afternoon all. Myself Jerome, and I'm in my 5th year of PhD under Rajagopal sir itself. So I work on Chlamydomonas, particularly characterizing few autophagy and with the interest to enhancement of lipid droplets.
Good afternoon, everyone. I am Jyoti Ranjan. I am working under Professor S. Rajagopal. Means like my work is mainly focused on Pison's and abiotic stress, mostly work on temperature stress and as well as fluctuating type stress. Thank you.
Myself, Mohammed Sohail. I am working on Rajagopal lab. I am just completed master's degree. Then I just work as a project assistant in our lab, mostly working on the photosynthesis and Pison's activa.
Kozami-san, would like to introduce yourself? Yes.
My name is Kazumi Takahashi. I am Yuichiro's wife. I was a school teacher, elementary and junior high school. But now I retire and I have a time to come India.
You are welcome. Okay, so Echiro, you want to say something or no?
I think I said already, but
yeah,
nothing to add.
Okay, so said this the workshop is about the both lectures and the tutorials. So every afternoon you have a tutorial, right? You can see the program. All of you received program, right? You would like to have physical copy? It's fine, huh? You want like to have physical 1 or 2 copy, you can make and give it to them. Whoever wants, you can collect from Jyoti or yeah, Jyoti will give you. So we have 24 talks, total 24 lectures and 7 tutorials. I guess 7 tutorials. No, 9 tutorials. Total 9 tutorials are there. Then except last day and all other days we have a tutorials. So each these 24 lectures. Now today we have 2 lectures, and tomorrow sometimes 3 lectures. And also Professor Raghavendra has taught, and as I said Professor Echiro Takahashi is a foreign faculty. I am also teaching some of the topics you can see, and here also we have invited Dr. Madan is going to teach, going to talk one of the topic, accumulation process. And there is one Professor A.B. Das from Utkal University who is also work on photosynthesis. He is here and he will be teaching 2 lectures, that will be day after tomorrow and afterwards. Day after tomorrow means tomorrow is 8th, 9th and 10th is going to teach 2 talks. And one more Professor B.C. Tripathi, probably in the program it is already there. So he is also expert in photosynthesis and chloroplast biogenesis in higher plant side. So he worked almost like same thing like about 40 years in the photosynthesis related area. So he is going to focus more on chloroplast biogenesis in plants and as well as the gene expression related to chloroplast. So 13th and 14th, 15th he will be with us. You can also interact with him. And one more, the other talks, my own student Dr. Yusuf Muhammad, who is going to take care of one, that is bacterial light arresting complexes. I also asked Jerome to take care of one of the talks. There is chloroplast transformation, one talk will be there, the rest I will be covering. So the main, as I said, the main motto of this workshop is to train all of you in photosynthesis related topics, training in hands-on training. So my students anyway, they don't need to train, but they will help you in training of all these whatever we have designed, and they will also be going to circulate all the the forms and on the not forms, the protocols. That protocols you can implement, you can go back and implement it in your lab. It works. So first we will start with the chlorophyll fluorescence. That is a simple instrument, handy P, how to measure the photosynthesis. That's a chlorophyll fluorescence. Based on the chlorophyll A fluorescence, you get the fluorescence, that photosynthetic activity can be measured. Then we also have is called photosynthetic, the PAM mission, that is little more higher end, where you can differentiate photosystem 1 and photosystem 2 and even cyclic electron transport. All these linear electron transport can be measured, linear and cyclic electron transport, that can be measured. That will be one day. And most of them, those in India and wherever, when they want to study some tree physiology, I mean tree related photosynthesis or higher plant related, they use IRGA emission. So IRGA will be also, you will be trained how to measure the photosynthesis, CO2, transpiration rate and CO2 uptake and water use efficiency and photosynthesis efficiency, and this also can be, you will be trained very well. Further, since it is the chloroplast biogenesis, and we will be going little deep into more of biophysical and biochemical experiments. You separate all the complexes like thylakoids, the first one, chloroplast and thylakoids. Then see what are all the complexes present in chloroplast. This very good one is called super complex separation by blue native gel. So that our students Pavitra and I think the names are there. I mean the new program, can you share with them? So all the names are there. Who are going to take care of the experiments. You can interact with them even if you go back and you want to get more information or some kind of help. They will be helping you. And now the blue native gel, and we will also see some biophysical aspects, how the protein and pigment they interact. So that using circular dichroism, only we will look at the UV visible side, visible range from 400 to 800 nanometers, that they will be also helping you to get trained. So you can train, actually now that is the good that there is only 15-16 students here. So you can have all of you can train very well if you are focused, I don't know if you are interested. So and now if time permits, we can add many more, but we will see. Then we will jump on to the one topic is metabolome, analysis of metabolome. That is using we have already metabolites already we have you measured, but you cannot measure now, it takes time. From that how we can analyze metabolites, either chloroplast metabolite, any metabolites, that will be Dr. Yusuf will take care of one of the chapter, one of the tutorial. Then true molecular biology, chloroplast to cloning, right? Cloning, cloning and transformation and isolation also will be studied. RNA isolation is also will be 2 days that will be also covered about the chloroplast and little more molecular biology. So these are the the experimental setup. So I request all of you concentrate on all those aspects, so that you will be really trained and it will be advantage for those who want to stay in this field. Of course, these techniques can be used anywhere, though unless, as long as if you are in a plant science, these techniques can be, you can apply, to check the physiology of the cell or physiology of the any leaf. This will be, really definitely it will be useful in future. So, and the sometimes these whatever we said, the strictly time, some time is meant, but even if it is late, you can stay a little longer and get you himself, you have to show your interest. None of my students will be more they will be active, if you don't show interest, they will also, okay, what is there, they are not showing an interest. So you have to show interest and they will be definitely work with you, or they will definitely train you much better. So we will also take help from Professor Echirosan if anything related, all these experiments. So that will be also, because you also can contact him anytime. You can get some information, any information related to whatever the topics which is going to talk, or whatever the topic, the tutorials which we are going to cover. You can also contact me or contact Professor Echiro. So these are some of the I mean, the kind of orientation I wanted to tell you before we go for tutorial. So we'll be using leaves as well as we'll also using cells, the chlamydomonas cells, to show you the function. Jyoti, going to use leaves also, right? Okay. Leaves as well as the chlamydomonas, so that you can understand much better the differential photosynthesis, if there is any difference in photosynthesis plant versus an algal system. If you have any queries related, other things, we are arranging lunch here and breakfast and dinner. Breakfast, when you come here, you can eat in downstairs, that you have to pay, because this conference, the workshop is a different kind of state. They don't give money much. They give only for for the speakers as well as some logistics. So that's why only some with the difficulties, we are arranging the lunch and breakfast, and dinner. You can tell wherever the guest house. If you are staying in the guest house, you have to tell them prior, so that they can prepare. Otherwise, these days you don't need. Swiggy is there. You can order Swiggy food, whichever food you like it. Any other things you'd like to discuss, or anything you want to tell all of the resource person for the tutorials? Jyoti? Pavitra, I want to say something? Not. Okay, so now it's like you have to feel like it's like your home, your institution. So all of you, so you have to be like friendly with all our students as your students, so that you can become good friends. That is very important. And after going here also, you should remember the workshop and also have that long friendship. It happened many times. They may come back for PhDs or very many interesting stories happened, but I don't want to tell all those stories. They got married also sometimes. So, so it's also it happens in many of the So anyway, so this is some of the you'd like to have some discussion, or you have any kind of doubts? Yes.
So the experiments which will take place in afternoon, like what is the maximum time?
No, that's what I said. As long you can stay long, if you want to close it, we will close it at 5 o'clock. Or you are from day scholar, that's fine. You can go ahead. You be there for few hours, then you can go. Yes. Travel, yes, I know. Where are you staying? Himayat. Oh God. Why don't you stay here?
Accommodation not available.
No, tomorrow onwards. No, you may get accommodation from day after. So that's you can get it day after tomorrow. I'll just tell message to tomorrow, because there is a convocation is happening. So all guest house are full. So by all of tomorrow 9:30 all of you join, because the inauguration is there. So after that you can attend classes. And one day I think Jyoti and Vijay, can you plan one day campus walk? Would like to go for campus walk? So I don't know, they know the places. Of course, I think I may know much better than you, but because I walk a lot. So White Rock or wherever place you design, and then we'll go. We'll follow you. I will follow Vijay, you, or you follow me? You follow me, or you tell me the place. Okay. Now I request Jerome and Jyoti, Jerome and Vijay, because he also does walk, always. So which day will be you can fix? Maybe no. HR also will join with us. This Sunday you won't be here. No, but one of the evening maybe you can make little experiments free, at least 2 hours, so that Okay, so how many are new to Hyderabad? Oh, you are also new to Hyderabad. Oh, you have visited many times. I think you visited Hyderabad? No, how many didn't see Hyderabad? There are places where Shilparamam is there, very close by. You can go there. You can have a good shopping. I mean, traditional shopping, traditional items, and all Hyderabadi biryani. We'll one day we'll have biryani. I already promised my students. So whichever biryani you'd like to have, we'll have biryani. Here only we'll have biryani. But you want to see Hyderabad Charminar, you can talk to my students. They can help you. Maybe the two we'll have one Sunday, right? One weekend, you can. Charminar also you'll have good the shopping, nice shoppings. Anyway, you talk to them. So, and many places to see here. So with this, I will stop, and if you let me know if you have any require any clarification or any doubts. Otherwise, we'll move to the lab. Jyoti will go to lab, or you will explain here about.
I have to bring the pots here if they want to explain.
Okay. Now, but initial you will explain. Huh? You will stay here.
I will just bring the pots from the plants.
Okay. So you will measure here only. Yeah. You want to ask something? So now. So once again, all the best for all of you, and thank you for joining for the workshop. and you will definitely will have good time here. And few more things, few announcements. The GIAN conference is a very prestigious conference in India. Not everyone will get this award. It's like kind of award for me to the department. It's like once they in a play, it is like we they scrutinize and review, and they select the project. And there also they have mandates that you all have to give attendance every day. I think Raghendra is collecting attendance, and there is signature, right? Yeah, you need to sign signature. That's very important. I don't know what they do if you don't come. I have no idea, but as a coordinator, I have to submit those the records, and also your feedback about the conference or the workshop. You have to give fair, your judgment. There will be a grading. You can give the grading. Those gradings also important. Improve from their side. Okay, if there is anything improvement, not paying money for these things. About subject, subject you need to be improved, or it could be much better, or some kind of anything improvement, so that they will improve, and then they will change the program slightly. So this is feedback, and attendance is mandatory by you guys. Okay, so all the best, Jyoti. Now you can start with the chlorophyll fluorescence, and you can measure yourself also. He will give enough time to you to measure, and also explain little bit the curves, OJAP curve.
We'll plot it in computer.
Yeah, you can do it there itself. Connect this computer. Huh?
Yeah, I can do it.
Yeah, here itself you can do it. Where is Shailesh? Shailesh is also helping, no? Oh, she went there to bring the part. Okay, then go and bring the parts.
Suhail already went.
Oh, there, it's there. Okay, then. So all the best, and I'll be in my office. If you, I'll in between and come. I will also see the results. If any problem is there, just give me, Jyoti. Yeah, you can close. Later you can.
So heat is majorly in the form of ATP. We are talking about plant photosynthesis. Got it? There are two photosystems, pH2 and pH1. So pH2 has a reaction center as well as pH1. So both pH2 and pH1 has reaction centers.
So, once the light energy falls on PS2 as well as PS1, these are all excited simultaneously, not one after other. Okay. So, once the electrons are excited, that chlorophyll molecules will be excited to the going to the excited state. Okay. Once it goes to excited state, it has to again come back to its ground state, and during that coming back, that energy will be released in a higher wavelength. Okay. The wavelength it will absorb will be a lower wavelength or higher energy. E is equal to h c by lambda. That means directly proportional inversely proportional to the wavelength. Energy is inversely proportional to wavelength. So, wavelength is less means energy is high, wavelength is more means energy is less. Got it? So, can anybody tell me at what wavelength region the light? 680 around. It exactly 680, it is from 400 to 680. Got it? And PS1 will be absorbing around 720, 730 region, that is it. Got it? That is why we call it PS2 and PS1. Why the naming is done PS2, PS1? Yeah, discovery of the PS1 was first, then came the PS2. Now again one thing, I told once the chlorophyll molecule is excited from basal state to its excited state, and once again if it comes to the basal state, that will release energy in the form of three ways. Okay, chlorophyll molecules. One will be through fluorescence, second one will be through heat energy dissipation, that will be your non photochemical quenching, and third one will be your photochemical quenching, that will be in the form of photochemistry. Photochemistry means your basic electron transfer, your cyclic and non cyclic phosphorylation, these are all your photochemistry. Got it? This is known as photochemical quenching, and heat energy will be again I am not going into details about different types of NPQ, but just for your knowing I am telling that NPQ is in the form of heat energy. Now, these three processes will be in a form of competitive manner, okay, not simultaneous. Competitive manner means if fluorescence is less, that energy will be taken over by your heat energy or your photochemical energy transfer. If photochemical energy transfer is less, that energy will be taken over by your NPQ and heat transfer. You got it? So, all three processes are your competitive manners. Okay. Now, what exactly this fluorescence, how does the chlorophyll fluorescence works? For this thing we specifically use an instrument that is known as handy P. So, basically this is a portable system. There is another system in which you can go into detail forms to check the fluorescence of PS2 as well as PS1. There are other instruments to check specifically PS2 portions only. So, why exactly we are taking this? So, basically this handy P, plant efficiency analyzer or photosynthetic efficiency analyzer, got it? So, this will only check the PS2 efficiency, not PS1. Why exactly is this? Can anybody tell me? Why this is just checking your PS2 efficiency, not PS1? See, I already told you PS2 absorbs what wavelength of the light? 680 around. PS2 680. So, what this emitter, this light emitter, this is a emitter, okay, and this is your component, the main box in which readings will be taken. So, here you see 3 light diodes are there, LEDs, okay, 3 LEDs are there. This will means this will give a flash of light, red actinic light, and this will be around 3500 to 3500 microingstine. So, basically the intensity of the light is known in the basis of microingstine or micromole per meter square per second, scientific terms. Thus, if you want to go into papers or research articles, they mostly prefer micromoles per meter square per second. So, the highest intensity will be around 3000 to 3500 microingstine or micromole per meter square per second. So, this light wavelength is around 680. So, can it excite PS1? No, because PS2 is only excited at that particular light intensity wavelength. So, this light wavelength is provided by this, understood? That is the main reason the PS1 fluorescence is not observed here. There are other PAM machines, next day we will see the PAM, and also 77K, I do not know we will be doing that or not, but for the PAM thing both light curves can be detected. For that it will use a far red light. The far red light comes under PS2, so sorry PS1, so it will excite the PS1 region, got it? This does not supply the far red light, this supplies the 680 nanometer normal light, red light, got it? That is known as actinic light. So, what does the actinic light mean? Actinic light, what is the actinic light? Actinic light means that light intensity which drives the photochemistry of a plant, got it? Photochemistry means you can say, like for example, the electrons are separated from PS2, initial charge separation is there, then after that it will go downstream, that means from PS2 to your cytochrome B6F, then to PS1, and again you can go to C3 cycle or dark reaction, or again you can go to cyclic reactions, got it. So, this is known as your basic photochemistry. So, that much minimum amount of light that is required for the photochemistry is known as actinic light, understood. Normally, if a plant is growing in actinic light, for example, we have grown this is a 14-day old pea plant, okay. So, it is grown under 150 microingstine. So, if a plant is grown in a constant light intensity, for example 150 or 200, and all of a sudden you are giving it a higher light intensity, shifting it to a high light intensity or again to a low light intensity. So, the first thing the plant will change is the chlorophyll fluorescence. The photosynthetic efficiency is the first and primary reason that the plant has to adapt, got it. So, PS2 as I told you, so excitation will be only of the PS2. So, there are three major parameters you need to see. First one is your FB by FM, FV by FM. FV is your variable fluorescence and FM is your maximum fluorescence, got it. Once we take the readings, that will just, this device will show your three values, F naught, FM and FB by FM. F naught is your minimal fluorescence, got it. FM is your maximum fluorescence and FB by FM is your what? PS2 efficiency. FB by FM is your PS2 efficiency. So, basically if a plant is not under stress, the PS2 should be, the PS2 efficiency should be good or bad? PS2 efficiency should be good or bad? If a plant is non-stressed, it should be good. Now, usually that good thing lies between 0.78 to 0.83, got it. Sometimes if it is, depends on plant, sometimes it might go to 0.84, but if it is less than 0, according to papers, if it is less than 0.78 or 77, then the plant is stressed, got it. So, basically this is the initial line of your check, initial line to check whether your plants is stressed or not. You can't directly take the leaves, go for everything and then check everything, that after that you get to know that okay, this is having stress, this is not having stress. The first thing you have to do is to go to HandEP, okay. The HandEP will detect your whether the plants will be in stress or not. So, if it lesser than 0.77 or 76 even, it depends on plants actually. Normally in higher plants the FBFM lies between 0.78 to 0.84. For algal sample, the highest FBFM might range from 0.75 to 77, okay, depends on the samples to samples. But for the plants I am telling, it can, for pea plants it should be 0.80, more than 0.80, fine. So, if it is more than that, then you can tell the plant is not stressed, and if it is less, then you can deduct that it might be under stress, okay. We cannot for sure say that it is under stress, we need to again do some other parameters to check, okay. So, this is the first line of testing to check whether the plants is under stress or not. So, the working of this instrument is very basic. This this emitter emits 680 nanometer of light that preferentially excites your PS2, okay. So, once the PS2 is excited, the light intensity is 3000 microingstine, 3000 to 3500, and that is very saturating, and that light pulse is very, means the duration of the light pulse is in milliseconds, got it. Now, the duration, if the duration of the light pulse is during milliseconds, why is it done? So, first of all you need to know about the PS2 and PS1, how does it work. So, initially when light falls on PS2 or PS1, the electrons are excited. After the electrons are excited, then PQA, that is reduced, then PQA to PQB, that reduction is there, then PQB to cytochrome B6F, then comes your whatever it goes on. So, there is reduction or not, donating the electron oxidation reduction is there, redox poise is there, right. So, if one is getting oxidized and the other one is getting reduced. Now, the thing is that already I told you the three things has to be competitive. What all the three things? What is fluorescence, second one is your heat dissipation, that is in the form of non-photochemical quenching, and third one is your photochemical quenching or photochemistry. Now, giving this saturating pulse basically inhibits your photochemistry, got it. Anybody knows about Kotsky effect? Kotsky effect, okay. So, what basically Kotsky effect states that if a plant is kept in darkness and again they are returned to the light, there will be a very initial rise in fluorescence and the rise will be exponential, got it. that will be exponential, and then there will be steady state of fluorescence. That rise in exponential, this part, that is represented in the form of this thing only, got it. That will be represented in a OJIP curve. I will tell you in detail how is it obtained and what exactly it means. So, basically what is the light intensity it is providing? 3000 to 3500 microingstine, microingstine or micromole per meter square per second, got it. One is SI, SI unit is micromole per meter square per second. Okay. Basically for paper publication we prefer micromole per meter square per second, got it. So, the meaning is same, you can use interchangeably, but for papers you have to write the SI unit. So, another thing is that, okay, so 680 nanometer will excite your PS2. Now, that reduction will be very quick, got it, because this is a very saturating pulse. Saturating pulse means when the reaction centers of all your your PS2, see, first I have to tell you before going into that, one is open form of reaction center, one is closed form of reaction center. So, open form of reaction center means when the PS2 reaction center is in oxidized state. Oxidized state means if the plant is kept under dark for sufficient amount of time, all the reaction center will be oxidized. That means no light energy is there, no electron excitement will be there, and all the downstream electron acceptors will be in oxidized state. They will not be having electrons to donate or to pass on, right. So, that is known as oxidized state, got it. So, that oxidized state gives a particular fluorescence level, that is known as initial fluorescence or minimal fluorescence, that is known as F naught, got it. Why you are dark adapting this sample? What is the exact reason? Just now I told you, the dark adaptation is done to open all the reaction centers. Open scientifically means oxidized state of reaction centers, got it. Oxidized state means all, there is no electron passing from PS2 to PS1 or anywhere, that means the plant has to be dark adapted, right, because the moment the light falls on it, there will be electron excitation and again there will be reduction, everything. So, we have to avoid that. That is the reason the plants has to be dark adapted, preferably around 15 to 20 minutes. For my plants I have kept a timer, so I kept it around 20 minutes, right. 20 minutes of dark adaptation is sufficient. Now, how the dark adaptation is done? It can be done two ways. Either you can keep it in dark room, okay, or else you can just wrap a black cloth around it, but make sure that there is no light. And third way, this handy P uses clips. So, clips will be clips. So, these are the basically clips. So, the work of the clips is just to dark incubate the leaves, not the algal cultures, these are for leaves. So, see here, you can keep the leaf here, there is a prong, you can open it. Open means while taking readings you have to, this is the closed form, got it. So, if you want to dark adapt a particular region, just clip it, got it. So, this one is not exposed, so that region will be under dark, got it. So, when you want to take the readings, just you keep this, close it, then you open the prong. If you do not open, the leaf will be still in dark and you will be getting different readings, got it. You have to open, then you can take the readings, got it. So, this way the plant, the area of the plant that the reading is taken is completely under dark, got it. So, this is supplied with directly here. So, this is any way of the dark adaptation. If you do not have the clips also, you can do it in dark room, or else you have to cover it with a black cloth, okay. Now, the dark adaptation time varies on species to species. Usually for my plants 20 to 25 minutes is more than enough. For algal culture 5 to 10 minutes is also more than enough, okay. Depends on what species you are working, got it. Now, this 20 to 25 minutes of dark adaptation will completely open your reaction centers, got it. That area reaction centers will be completely open. Open means in oxidized state, got it. So, now when I apply a flash of light, what exactly happens? What do you expect that should happen? If a dark adapted leaf, you are applying a flash of light, that to be 680 nanometer, that is exciting the PS2 preferentially, not PS1. So, what exactly should happen? Can you correlate? There will be reduction, okay. But how fast the reduction will be there? Because the time, this pulse is, the pulse time is in milliseconds. It is very short pulse and at very high intensity. So, what it will do? It will suddenly reduce all the reaction centers. Got it? Already it is in oxidized state. Once you apply the pulse, it will be completely reduced. So, that reduction will be there. For example, this PS2 reduction, initially the QA reduction happens. So, all the QA are getting completely reduced all of a sudden. So, it will take some time from QA to QB to take out the electrons, because the reduction is very sudden, right, because the pulse the duration of the pulse will be in milliseconds, very high intensity and very less time the pulse will be there. Got it? So, that pulse will reduce your reaction centers. Reduce means the reduction will be very quick. So, if the reduction is very quick, that means what? Basically, why saturating pulse we are giving? To inhibit photochemistry. So, inhibit photochemistry means we do not want the electrons to pass from PS2 to PS1. That is the reason of saturating pulse is giving. So, we are just overwhelming the PS2, right. So, overwhelming means PS2 it was already in oxidized state. So, if a sudden flash is given, all the reaction center will be suddenly reduced, and that reduction it will take some time to again re-oxidized. Okay. That is the reason we are using a saturating flash. Saturating means normally 1500 to 2000, even 3000 is more than enough saturating pulse. Normally for your sunlight during the highest hour it will be 1500, 1700 microingstein. So, 3000 is like double to it. So, this saturating pulse is given to reduce all the reaction centers. So, initially it was in completely oxidized state. Now, after applying the saturating pulse, it is completely in reduced state. Got it? So, there will be a basal level of fluorescence when it is in oxidized state, and once the pulse is applied, there will be complete reduction fluorescence. Got it? So, basically there are two readings, one is F0, one is FM. F0 is your minimal chlorophyll fluorescence, got it, and FM is your maximum chlorophyll fluorescence. Got it? What does FM means and what does F0 means? So, that represents the F0 and FM is basically in the form of a OJIP curve. I will show you once I take the readings. What does OJIP curve, what does O to J, J to I and I to P transition means? Okay. That will be in milliseconds. That means the plotting will be in between 1 second. Okay. So, light will be from 0.01 milliseconds to 1000 milliseconds. 1000 milliseconds is basically 1 second. So, in between that the fluorescence will be there. So, this is basically the Koteski effect. Got it? Koteski effect is, if already I told you, there will be a steady sudden rise in fluorescence level once the plants are illuminated with a saturating pulse. And after, if you want to check whether the fluorescence level, so if I ask you a question, if the fluorescence is accumulating or there is a exponential increase in fluorescence once the light is applied, after 1 second what do you expect? So, after 1 second what do you expect? There will be rise in fluorescence or decrease in, or there will be steady state? Just think about the normal photochemistry. So, what I asked you, initially you kept the plants under dark, right, then you applied the light. For example, I applied light with this one. So, within 1 second there should be a initial rise in fluorescence, that is the Koteski effect. After 1 second, what is the consequence of that fluorescence? What should happen? Basically decrease. So, will it decrease to 0? So, it will not decrease to 0. There is a reason for not decreasing to 0, because the plant still emit of minimal fluorescence. No plant gives 0 fluorescence values. Got it? So, there is a F0 and that is the reason there is a F0 and there is a FM. Got it? So, once you excite the chlorophyll molecules, there is a OJIP curve, fluorescence in the form of fluorescence, and that fluorescence, if you will continue for 5 or 10 minutes, same taking that fluorescence reading, if you take it, after 1 second you will see a steady decline and then there will be a steady state. Got it? That steady state is because of activation of what? Let me again tell you, initially you applied light, got it, the reaction center were all oxidized before application of the light. Now, you applied a sudden pulse of very high intensity, got it, then again from oxidized state to completely reduced state, F0 and FM is obtained, right. After that what do you expect? So, already I told you pH2 reaction center will be oxidized and again pH2 reaction center will be completely reduced. So, there are electron carriers beyond pH2 or not, right? There are pH2 carriers, so electron carriers beyond pH2. Now, after 1 second, that electron carriers will start taking electrons from the reduced state of pH2. Got it? Reaction center, from those reaction centers PQA, PQB, cytochrome B6F, all these will be taking electrons slowly. So, basically if those things are taken slowly, so fluorescence will drop. Why it will drop? Already I told you in the starting of the experiment, all these are competitive process. Okay. So, saturating pulse you are applying to inhibit photochemistry for few seconds. Within 1 second you are inhibiting the photochemistry. So, if photochemistry is inhibited, that energy will be taken over by your fluorescence. So, fluorescence will be detected by this. It directly does not check the photochemistry. Got it? So, photochemistry has been detected through fluorescence with this machine. Okay. No instrument detects direct electron transfer. Electron transfer, they will inhibit the electron transfer and that energy whichever is taken by the, you can't, obviously you can't like NPQ form of heat dissipation, you can't, there is nothing in your hands, you can't do with the NPQ, but you can definitely inhibit the photochemistry. So, once you inhibit, the better the fluorescence, the better the plant performance. Why? Because the photochemistry, if the photochemistry is good, the amount of, if you inhibit that much amount of photochemistry, that higher photochemistry will be reflected in the form of higher fluorescence. Got it? If a plant is having lower photochemistry, if you inhibit that photochemistry, that lower photochemistry will be reflected in the form of lower fluorescence. Got it? So, these are all the basics of your chlorophyll fluorescence, be it dual PAM or be it this handy P, right. So, dual PAM will be a much advanced thing. So, tomorrow we have to do dual PAM. So, starting with anybody has doubt regarding this, rest of the things I will tell you once I take out the files. Okay.
Both the principles are same, dual PAM and
dual PAM, yeah, dark incubation, it has to be done for everything because you have to open all the reaction centers. Open means oxidize, okay, completely oxidize all the reaction centers. Otherwise, you can't know the what is the basal level. You got it? For example, you took this plant, already it is growing under a particular light. You directly took it for fluorescence. So, it is already under a photochemistry, right? The electrons is getting, and that is why it is growing. So, some of the reaction center might be in closed state. Closed state means in reduced state. That means some of the reaction center will be actively engaged in photochemistry. So, you can't know the exact amount of reaction centers that is actually present. It will just detect your open reaction centers, this machine. So, the dark adaptation is very much essential, got it. But for PAM, the adaptation is same, but the working, the fluorescence, pH2 fluorescence is same, but coming to the light curve and everything, I will explain tomorrow how exactly it is done, but the basics are same, got it. So, so it is almost over, people can come here. So, see, this is the main console, got it. I think some of this is very basic instrument, whoever works on chlorophyll fluorescence and all, they have this. This is the primary instrument to check the pH2 performance or the overall performance of a plant, got it. So, this is the console and this is the light emitting diode, okay. This is the major emitter. So, 3 LED diodes are there. So, this will emit the light pulse, I will show you the light pulse, how exactly it is done. So, just check, see. So, this is 680 nanometer light, got it, red light 680 nanometer. So, red light basically activates the pH2 properly, okay. So, that is why red light, some nowadays many PAM machines or NDP are coming, they use mixture of blue and red light. This is the older version, that is why it uses a red light, but irrespective of the light they use, the excitation wavelength should be 680 nanometer, got it.
It affects that
no, it does not affect because I have already dark incubated. This thing is not affected with this light.
Okay. Exclusively.
Yeah, these clips are exclusively fitted on the surface of this one, so that no stray lights are there. So, you can, if you have the clips, you can perform it in a well lit area also. Okay, you do not need a dark house or anything. Got it? So, this red light is 680 nanometer, it will excite your PS2 only, not PS1. Got it? So, if you excite your PS2, now next thing I am showing you with an example. Okay, this is the example I am showing. See here, first recording will be in file numbers. So, depending on the number of files you are saving, the file numbers will be there, file ID you do not need to tell. So, F naught, there will be a value of F naught. I already told you minimum fluorescence, if you do not adapt, then the F naught value will change significantly. So, you should dark adapt it, right. Then comes your FM. FM is maximum fluorescence. Maximum fluorescence will come after this light. Basically, all these readings will come after this light application only. Then comes your FB/FM. So, this is your major thing. So, if you want to check exactly how a plant is performing, you just check the FB/FM. Got it? So, if you want to get a lot of plants, people grow in fields, right? 50, 20, I mean maybe 5 acres, 10 acres. So, you can't go individually collect each plant and all. So, they will just take a sample. They will just take, yeah, random, they will take a sample. That random sampling can be done in this one, right. So, you just take, it will take hardly 1 second, 2 seconds to take the FBFM readings, then you can later analyze it. Okay. Analysis part I will tell you how it is done. So, I am showing you again. So, this is a cord. So, basically it is like a LAN cord only, you just need to connect it, then just switch it on. So, after everything is loaded, this thing will show up. Okay. Measure. Another thing, before starting experiment, if you are switching to different samples, you should always do this. Just go to PC mode, okay, this one, sorry, system, then you need to go to configure, then you need to auto gain. Auto gain is basically the plant, means the plant's this instrument's automatic function, it has defined something, so that for each sample it will, according to the chlorophyll concentration, it will make up the light intensity accordingly. Got it? But that light intensity will be a saturating pulse. Got. So, here I am just taking. So, before auto gain you have to clip this, otherwise it will not work. So, you just clip it here. Another thing, make sure that you do not plug the leaves. If you will plug the leaves, the FBFM will change drastically. If not, maybe for my samples it may not be different, but for other leaves it might be different. So, IRGA, dual PAM, handy P, if it has to be performed with the in vivo experiments, it is good. If you can't perform, in some people they use it inhibitors for the leaves. So, in that case you can't do anything, you can take the leaves directly, but if it is possible to take the readings in vivo, then that is good. Okay. So, second thing, we have to remove this. Got it? So, before taking readings we have to remove. Now, the area of the leaf is exposed to the light source. Okay. The light is not applied yet. So, there is no reading. So, first you have to do is auto gain. Okay. So, just click here. So, auto gain is detected. No.
F naught value it is there.
Yeah, F naught value, no, I just, for showing purpose I just done, that value is not proper. So, once the auto gain is there, there will be some gain number 1.1 or 1.2, 1.5 depending on your sample and chlorophyll ratio, that detects automatically. So, you do not need to do anything here. So, then you need to go to measure. Okay. So, if you will measure, so the light pulse, it will go inside it. Now, come here and see. So, so apart from file number, see the F naught 563, FM is 3671, FBFM is 0.847. So, it is more than my plants are performing proper. For me, my control plants will be having this much values only. So, if I get a stress, the value will come down to 0.6, 0.7, 0.5. If it comes below 0.4, it is under too much stress. The plants cannot recover after, if it is below 0.4. Got it? So, this is basically, so first thing you need to write is the file number. Okay. Just write the file number 346, then write the FM 3671, FBFM 0.847.
You have to minus FM minus F naught.
Yeah, FB/FM is basically, FB is variable fluorescence, that is FM minus F naught, but since we have done the auto gain, yeah, you can take. So, since you have done the auto gain, it will not exactly show your values of the FM minus F naught. If auto gain is not done, then you will usually the value will be same Fm minus F naught divided by Fm, got it. So, Fb is equal to Fm minus F naught, but we did auto gain. So, it will add and deduct some values from the machine side, got it. Yeah, it depends on your plants. Usually if you are changing samples, now I am using pea plants, then I want to compare it with a completely different species, exactly, right. So, there the chlorophyll content might be more or it might be less also. Yeah, no, light wavelength, it does not matter, it is a inherent capacity of this instrument itself. I do not know exactly how it works, but auto gain has to be done when you are changing samples. If you are using the same pea plant in a different setup, or if you are taking biological replicates of pea plants or even technical replicates, that is fine. But if you are taking pea plants here, then stressed kind of pea plants, then you have to go for auto gain for that sample, got it. So, once auto gain is done for this sample, you do not need to do it again.
Here you can change the light. If suppose PS2 is 680 like that.
Yeah, no, light intensity is fixed, sir. You cannot change in this one. But PAM, it can, PAM is basically dual PAM. So, there is a dual wavelength of light. It will excite the PS1 as well as it can give the far red light as well as normal red actinic light. So, that is more advanced, that is a big one. So, you can't carry it. This is just for initial thing to check PS2 efficiency, just for field purpose. So, if you are getting proper stress, if you show there is a lesser value of 0.8, 0.72 or 0.68, even 0.75, then you can claim that it has some stress and you can go for further things, right. So, you have written these things.
0.8 whenever value come, that is under the
according to no, according to some papers, actually it depends on species also. For pea plants, if it is coming less than 0.80, it can be regarded as stress, but not too much. 0.6 is considered as a ideal stress, 0.6 to 0.7. If it is 0.5 also something you can do, but if it is coming 0.2, 0.3, 0.4, it is under too much stress. Even if you try to recover the plants, it cannot recover.
Stress in the sense any water stress?
Any kind of stress, any kind of stress, any kind of stress.
Regarding to literature?
Yes, literature states that if it is 0.77, I will talk according to literature only. So, if it is 0.77 or higher than that, then the plant is not considered under stress. Okay. So, that also depends on the samples. This is my P sample, but for algal sample, the maximum FBFM will come 0.76 or 0.72. Okay, and the minimum might be less also. So, that depends from sample to samples, got it. So, you wrote these things. Okay. So, these things you do not need to write TFM area and all. So, basically I will tell you what does this means. FBFM you wrote, FM wrote, main thing is this file ID, right. Okay. So, once you put escape, it will be saved automatically, got it. So, second time you just press it again, it will come back to home screen. Now, you do not need to do auto gain for the next sample, because it is under same set. If you are changing the tub or a different set is used, then you have to go for auto gain again, got it. Because if we say that the plants are growing under same conditions, but technically what is happening, what biologically is happening, we do not know that thing. So, that will equalize the chlorophyll more or less, right. This auto gain will equalize more or less the chlorophyll, and then it will see the pH2 efficiency, how is it getting affected, got it. So, anybody wants to do, you can, yeah, you can do one by one. Just make sure not to click this. If you click this, then it will trigger the flash. I will show you. See, so this is actually a manual method. So, you should not. So, you see, FBFM is high because no leaves are used. So, 347, no need to save.
Okay, every experiment.
There is also manual saving also. We can also deactivate it, but for lab purpose, I am not deactivating it.
First we have to push, no?
Yeah, here.
Back.
But make sure the leaves are not detached. Okay. If it is detached, then
we won't get lucky. Now
no, you will get some values, but that values you may not rely it completely.
Yes. Now we have to open this.
Yeah, open it.
Then we have to give.
Yeah, fine. So here you see 0.843. The first reading was 0.847. So, because these are grown in same pot, it will more or less have the same
Can I
readings.
switch off this one?
Yeah, it's done. Escape, you can take it out. So, if you will take again, once the readings has been taken, if you again flash the light, the BFM will decrease. Got it? Because already it is in excited state, and again you are re-exciting it. So, already the plant has received a saturating pulse, and again the saturating pulse is applied on that. So, plant is coming under stress. So, that will show a
Can you do that also one more time?
you can do. I will show you here. Clipping process or autogen process?
Autogen.
Okay. So,
This is handy piece, sir, actually.
yeah. So, first you clip it, then go to escape. This is the main menu, right? You just go to your system, then go to configure, go to autogen. So, make sure this is open, right? The autogen came same around. So, just you again press 2 times escape, it will come back to main menu. Okay, then you can go to measure. It will come 0.84 around something. Yeah, 0.847. So, you can try if you want. Just a minute. Saved it 349 because we need to extract those files also. Yeah, you can remove now.
So automatically measuring or it
Manually they are pressing.
by this one.
Yeah, you can remove it. So anybody else wants to try?
0.82,
350. 350. Just write the file name 350. File name should be enough. I wrote it just to cross check everything is fine.
Okay, how do I save it?
Escape, escape. It will come back to the file saved. Okay. This one the automatic saving is switched on. That's why it's saving. Otherwise, before once you press escape, it will ask whether to save file Y or no. So that way you can do. But for our lab purpose, we have just. Let it complete rest of the thing. Okay, 0.854 around that same it will come. 351. Just write down the file. Yeah, algal also I'll show you how it's done. Algal cells also, but the setup is a bit different. You can't use this clips in algal thing. It, don't press on this, otherwise this will loosen.
I think leave is
no, fine. Fine. 352. Normal, not 0 point. That's the file names. Okay. So you can detect once you take 5 or 6 readings, you will be getting to know that what is the average BFM that's coming, right? So if the next leaf you are taking and somehow the leaf is turned over and you clipped on or something, there will be a drop in BFM. That way you can know that it's not proper. No, you opened it, press it again.
It will be
no, fine, 0.39. You pressed first, might have been 0.
It will come like this.
So if you press like this, the problem is this fitting thing. If you hold it here and press, the leaf will dislodge from here, and that will be exposed to light. So you need to do it properly. You need to hold it here, then you need to open. Okay. One more is there. After this, we can switch over to algal cultures. I will show you how it's done. There. Gone. Okay, it's gone then. Once it is exposed to. Yeah, you can use whatever the minimum leaf area is this much, this inner hole. Yeah, see this, once you have to open this leaf first, you need to see which area of leaf you are exactly taking, and just go to a leaf. Once you clip it here green, okay, and close it, then close it, done. You just wait for 20 minutes or 25 minutes, it should be enough. Got it? So now we will go with algal culture, how to check. So you have written the file name, right?
Till 352 you told me.
I just write 354, right? Last 354. I think 353 also it's there. 353 is also there. 3 and 4. After this you also write 354.
So we have to expose the light only to the upper surface of the leaf.
Yeah, it should be done. This is the dorsal surface of the leaf is basically this one.
Yes.
Okay, this is the ventral surface, so it has to be on ventral surface. Okay. The part that is exposed to the light. Okay, this is I think adaxial surface, the first one, top one. Yes. This is abaxial. So adaxial should be exposed to the light surface. Leave it. We will do with the algal culture. You keep it back.
How long you will take by?
15 minutes.
So F0 is before exposing it to light, or
yeah, F0 is a baseline. There is actually the thing is that before measuring the ABFM, this instrument gives a very slow light intensity, that is known as measuring light. I didn't go more into details. That measuring light will be like 10 or 5 microingstine. So that basically will determine your F0. Okay. So after that the flash will be there. Okay. Vijay, algal culture. Dark incubated?
Are you showing the graph? graph? Need any pen drive?
We need. Pen drive is not needed. My laptop is connected, na? You can show?
Yeah, I'll get that.
You show, and that time I'm ready.
Why you are taking more lambda models? Why not other?
Salmon is a model of
Both is our
Vijay, where is Vijay? He's coming.
Hold it, give him.
Yeah.
You're going down or you'll come back?
Yeah.
So by that time I'll transfer the files. Yeah, I have to plot it. There is a specific program to open that thing. So once it is your readings are done, you just name the file numbers. File numbers is enough to cross check whether your file numbers is same. The problem is that if somebody comes and does after you and deletes some file previous to you, your file numbers will be shifted from 53 to 54. Or sorry, 53 to 52. Got it? So it's better to write your file number. Since I'm as long as you take it after you record, that's fine. But if keep it some 5-10 days and somebody uses it, the file numbers will change. That is the reason I always have file and ABFM values. That way I can cross check whether all the readings are proper or not, right? So to take these things readings, first you need to this is a connector, normal connector. So you just need to connect to one side.
No, not required.
You can come here just to see. It's fine. No, I'm just transferring it.
Bag lo
This is a normal USB.
దీంట్లో లైట్ వస్తుందా
రెడ్ లైట్ పడుతుందమ్మా రెడ్ లైట్ వస్తుంది కానీ 1 సెకండ్ మిషన్ కనెక్ట్ చేసినప్పుడు యాక్చువల్లీ ఇది డార్క్లో చేయాలి కదా
ఆల్రెడీ ఇంక్యుబేట్ చేసినాడు
కాల్ మీ రైట్
ఇంకా టైమ్స్
అగైన్ బ్రింగ్
But we don't use that because this is a newer version of NDP. The older version NDP uses
bit of light. That's fine. Just for
demo.
For experiment, yeah. Don't forget.
Don't forget it.
Communication.
My turn.
So first you go to handy P, then select the COM port. Okay. So only one communication port is detected, so you can just select it. Go to OK. Got it. Then you have to go to handy P, then download the recordings. So tell me the first file number. First file number 3. Yeah, 346 only, that's it. So the last file number is already displayed, you don't need to change anything. So if you have some problem like BBFM was saved, you didn't take proper readings and the readings are saved, you can check over here. Got it? It will export all the files from 346 to 354. Just go to OK. Okay. So these are all the readings of all the whatever values we have checked. So our most of the values came 0.84 around, right? But you see here one red color is coming, right? Red dot. The ABFM value is 1 because we didn't place the leaf. We directly took the readings. That's why this values, this indiscrepancy came. So first you have to you can either file exporting, you can delete the file later on, just you remember which file number, because during export the file number will be also same. Got it? So if you will just see the graph, so this is your OGIP curve. Got it? This is from 0.01 millisecond to 1 second. Got it? 0.01 millisecond to 1 second. This is in microsecond, that's why you are seeing this. Okay, if you convert it, this is basically 1 second time. So this is your the least one, the down one you are seeing, na? The down, this point, dot dot points, this is your the red one. Okay. So you can take the average of all these things. So you can see properly 3 humps. Got it? First hump, second and third. Got it? Another thing I told you, after 1 second or after 200 to 300 milliseconds, the fluorescence starts to drop. Why is that? Because the photochemistry starts to kicks in. That means your downstream electron carriers will be again taking over the electrons. If they start to take the electrons, that fluorescence will decrease. Got it? After this, I am telling you, after 1 second what exactly happens, this drop happens and again plateau phase happens. Okay? This is known as that plateau phase is known as steady state fluorescence. Got it? So this is your O. O is your minimal fluorescence. Got it? Minimal fluorescence after projecting the measuring light. Measuring light intensity is very less light intensity, 5 to 10 microingstine. Got it? After that will measure your F naught. Got it? Then this is the O phase. It normally starts from 0.01 to 0.05 milliseconds. Then this one will be your J. Okay? J will be coming around what point? You just see here what point it comes. 2 to 3 millisecond it should come. Got it? 2 to 3 millisecond it should come J. Then I will come around 30 milliseconds. Got it? And P, this P is highest fluorescence. It's not at this point. highest fluorescence is at this point, got it. After this it starts dropping, so you can't take this as the highest point, it should be taken at this point. So it will be around more or less 200 to 300 milliseconds, got it. So this part is 1000 milliseconds, 200 to 300 milliseconds will be your FM or P. P is your peak, got it. Peak means highest fluorescence amount, that is FM. This is your peak, this is your I will be around 30 millisecond, then here it will be coming around 2 to 3 milliseconds will be your J, and this one will be your O. So O to J represents the initial QA reduction, got it. O to J will represent your QA minus formation or QA reduction. Then OJ, J to I will represent your QA minus to QB minus formation, got it. See, all these things are getting reduced. Then and also J to I will also represent your initial reduction of PQ pool, okay. First O to J is your QA to QA minus formation, got it. J to I will be your QA minus to QB minus formation, then I to P will be PQB to PQB minus, and as well as the partial reduction of the PQ pool, okay. And your P will be, or I to P will be your complete reduction of the PQ pool, got it. So from here to here you see it is going up to PQ pool only, it is not going beyond that, got it, because this is all related to PS2. That is the reason it is not going to PS1 region, okay, because we are giving that much light intensity only, 680 microangstrom, got it. So go to that. This graph is not needed much. This is fine. So these are all your parameters. So parameters is used to plot your radar plot and everything, okay. So you can directly see the parameters if you want to check. Apart from FB FM, there are many other values. F0 FM values you already have, FB FM values you already have. Apart from that, there are other values. You can go through it, and if you want to plot it, that is usually plotted in the form of radar plot. I am not going into detail because the major thing is these three things, got it. Next, just check. Okay, leave it. Okay, after that how to export all these files, you just again you go to the summary part, got it, then go to file. After you go to file, see there are three things to extract. One is your parameters, one is data points, and one is hand file. Hand file means handyp files. It will be saved in the form of handyp format, okay. You can reopen that file again in this software, got it. So export parameters I showed you, apart from FB FM, F0 FM, there are other many things parameters, you can export that thing also. Data points is important to plot your OGIP curves, okay, and OGIP curve is always plotted in the form of exponential curve, got it, not normal. Your x axis will be your exponential curve, okay, not linear, it should be exponential. That way only, yeah, log phase, log 10, that way you will get all the three things, otherwise you will get this kind of thing, this graph you will get, okay. So just export data points will, only data points. Parameters I am not showing because you do not need that much to get into parameters, but data points I will just save in desktop. Okay, close it. No, got it. So open that thing. So once it is done, these things you see, these are all your record numbers, 346 to 354 file numbers. Record numbers is your file numbers. This is all exported into Excel CSV format. So this is your in seconds, this is not microseconds, this is in seconds. So you have to convert it into milliseconds. Millisecond means you have to just multiply with 1000. But okay, so this one will be your 0.01 milliseconds, and the last one will be your 1 second. Sorry, 1000 milliseconds. So we'll do this once. Yeah, you can delete all. So top things you can delete, that is just the exported parameters and everything. So, what you need to do, you have to superimpose, you have to go to transpose it. Got it? This is a transpose. Delete this. This was the odd value we are getting. 347 was the odd value. You see here 1 0 0 0 0 2 0 other things you see, these values, just delete. Delete. So, here you see up to 1 only the readings are there. 1 means up to 1 seconds. So, you have to multiply na 1000, right, 1000. So, record number you have to avoid this, do not confuse this with the actual value. This will be your record number or file number. Okay. You can identify with your these values, there is much difference in it. So, you have to just multiply your seconds to 1000. So, it will be in milliseconds, got it. 1000 into this value will be your milliseconds. So, 0.01 millisecond, and if you will drop it down, it will go to 1000 milliseconds. So, you can write it time in milliseconds, go down once, delete other things. See, up to 1000 the value is there, right. So, 1000 means like 1 second here, and here it is till second, got it. So, now you have to take average. All these things are from same area, right. So, if you want to take technical replicates, you can take all these things. So, you just start from here, you go to here, then you can take the average. Usually in OGIP curve you do not need to plot the error bars, because you take too much of averages. Okay. So, you do not need to plot error bars. No, not that, this is file number. Okay. So, this is your average file, got it, and you just write CT or whatever the name, if you want to have a control value or wild type, whatever you need to write, you can write it, then just select these two until 1000 milliseconds. You look at here, it will be up to 1 second only. Okay. So, this is in till 1 second. If this is what
is going on.
Then you go to insert, just plot this one. Okay. So, this is non-logarithmic scale, this fluorescence is non-logarithmic scale, that is very different from the one you saw earlier. Okay. Now, how to change it? Just click on x axis, go to your logarithmic scale, log base 10. Now, you see it came, the values came exactly how you opted. Okay. This is OJIP. Okay. So, this is how exactly it is plotted. So, you can compare the OGIP curve of one with the other OGIP curve. You can just superimpose all the images and see whether the fluorescence is high or low. The better the fluorescence, the higher the FM, and some F naught may or may not increase, depends on your stress, but F naught, if it is less, having lesser above FM, the maximum fluorescence should drop. Got it? Okay. So, you have any doubts regarding this plotting? If you do not have, then algal samples you can start. Fine. Close it. Take this. So, Vijay will show you about the algal samples. The incubation, the basic working procedure is exactly same, just the incubation way and how it's incubated is just different. Okay, and how it's how the readings are taken.
So here the mechanism is same for both plant and microalgae. So here I have taken the culture, that is Chlamydomonas reinhardti. It's a photosynthetic microalgae. So here also you can measure F naught, FM, Fe by FM in this photosynthetic microalgae. So I have just dark incubated this for 20 minutes. So this experiment has to be done in dark. Okay. Since for demo I am showing in light. So this is the algal culture I have taken. So the parameters is same, just switch on this. So as he said, first you need to go to system and then configure, but before autogen you have to load the sample. Okay. Unlike in plants, you just have to take here, you have to use this long tube. Since it's a liquid culture, you need to add like 1 ml. Okay. So first you need to wash your tube and then you need to load your sample. So you just have to place your tube in this. So 1 ml is efficient. So first you need to do autogen. So after autogen you just discard this. So now measure. So here Fe by Fm it is showing like 0.584. So unlike plants, it's in plants it shows like 0.8 or something. So it differs in microalgae. So 0.7 is efficient.
Difference you can see that for plant and algae.
Yeah. So you have to compare in microalgae itself. Like plant it will show high because microalgae depends upon the stress conditions, light parameters and everything. So maximum we get here in our lab is 0.7, above 0.7. If there is any stress like abiotic stress, if you give highlight treatment or high temperature, the Fe by Fm value drops. Like it will show around 0.4 or something. So usually it should show 0.7. Here it is showing 0.5. It's a little old culture, so I'm just showing for demo. So after noting the values like file number, F naught, Fm and Fe by Fm, you just have to save it. Okay. Save file, yes. So likewise you have to do all the samples. Usually we will do like control treated samples. So we treat our culture in highlight temperature or high temperature. So we will take two samples. One is control, that is without any treatment. One is treated sample, that is stress we will give to that sample, and then we will just compare with control and treated. So those values will measure and will plot same. The procedure is same. You just have to export the files and just compare like how it is differs from control to the treated. So this is the sample.
So no need to press anything here.
No, nothing.
Just like we did with.
Yeah. You just have to put your sample and just measure. So anyone wants to do or
Yeah.
you want to do?
Discount?
Yeah, discount. You can take that.
System is different. System of NDP is same, but
yeah, you just have to put this top layer just to hold the test tube. That's it. But the light diode will be same. 3 light diodes will be the same. No, keep this. It should be done in dark actually, so we'll keep that.
Measure.
Since it's the same sample, you don't have to do autogen each time. One more time. Okay. Yeah.
0.598.
0.598. So you have to take triplicates and you have to take the average value and then you have to plot it.
So triplicates means every sample.
Yes, if you have many 2-3 samples, each sample you have to take 3 readings. So 3 times you need to take and 3 times you have to measure and you have to take the average values. Anyone wants to know? Measure, okay. 0.58. 88. Usually these algae will grow in 3 days. So third day it will show the maximum Fe by Fm ratio.
Yes.
If you want to save, you can save yes.
Okay.
Anyone?
But for plants we were pressing the button to expose it to light. But for algae it is like some auto.
The light itself comes. When you press the measure, see.
Okay.
It only measures when you press the measure. Green one is algae, microalgae. So this is Chlamydomonas reinhardti species.
Okay.
Yeah, press OK. 0.615. You just have to check Fe by Fm.
615.
So this is the procedure. It's same for plant and microalgae, but here in this case you have to put the tube and just have to measure 3 readings. In plant you have to keep the clips and everything. So this is it. Any doubts? You want to know anything? You want to do? Yeah.
We expose it to some stress. Will the Fm increase or decrease?
It will decrease.
It will decrease.
This is last file, so yes. Then measure. Okay, click OK. Yeah.
2.6. Have to wipe it with tissue.
Yes, if you touch with your fingerprints or anything, just to remove that you need to. It's always preferable to wipe and do.
Yeah. Sir, if we redo the sample like without scarring.
So it's okay now?
Yeah, press OK. One more time. Yeah, measure. Okay.
Sir, if we redo the sample without scattering, will it show any pattern?
It won't show much difference. Maybe in 0.1, 0.2 difference it will show. But if it's the same sample, if you are taking multiple time reading, it should show similar readings. Yeah.
If we keep the same sample and like measure it
Do
once
multiple
more.
times. Yeah, it won't show much difference. It will show. You can just try it. Yeah, you can. 2 times you click. Sometimes you'll forget to save the file. In that case, what you'll do? You have to do again.
I have a doubt.
Yeah.
If the alveolar sample is kept in stress, then what will be the reading?
Reading will differ. Like to check how photosynthetic efficient it is, you need to measure this. So if the value decreases, there means some stress. Like initial chlorophyll, this Fe by Fm drop means what is happening is plant is not efficient. Like photosynthetic efficient, it is not there. So there is a drop in Fe by Fm value. So you may think like since we are giving stress, it's not performing well.
What is the range of the reading that will come if it is in stress?
It depends on stress. Like there will be medium stress, high stress. So if you keep if you subject your sample to high stress, there will be a much difference.
For good condition, what will be the rating?
Like in normal conditions, 0.7, 0.7 to 0.8. Usually this sample, this culture, this chlamydomonas usually grow in 3 days. So third day it will show maximum Fe by Fm. You can check daily wise. Okay, it will differ. It will increase gradually. It will increase. If there is any stress, there will be a drop. So it's like preliminary test that you'll do, how it's performing.
How is the F0 value is being calculated there? Can you say?
How if not?
F0 value is how calculated? Like when it is calculated?
See what happens is you will dark incubate your sample, right? So what happens in dark conditions, all the reaction centers in PS2 will open. So it will wait for the light. So there is a stress that dark you are giving. So all the reaction center will open. If there is a small light pulse you will give, it will take the fluorescence. So the fluorescence that is emitted, it will show in the form of values. Those values will tell you if it is in good condition or bad condition. Like if there is a high stress, this chlorophyll A molecules or the PS2, if there is a disoriented structure is there, the difference you can see in the values, like there will be a drop or something, then you can say okay, there is PS2, some disorientation is there structure. So to confirm this, further experiments you have to do, like BN page that you will be doing.
The FM value is when we put the light and it absorbs the energy and
it excites the electron flow is yeah,
it reduces to
yes,
give the
yes.
FM value.
So
Like that,
yeah,
what is the F0 value? Means how
F0 is like minimum fluorescence, where the whatever the cells are there, minimum fluorescence what you will give initially, that is the F0 value.
When we are giving initial fluorescence?
Initial fluorescence is the sense you will give the light pulse, right? Red light you see, you saw that. When you kick the measure, it's a process, right? The curve will be like this, right? O is like OJP transient is shown, no? OJP curve. So O is like F0, initial value. Maximum you will get when it is exposed to the light, like the time will be there.
Like what is the threshold value that is already being present?
Yes, very light.
So though
Yeah.
it is in dark,
Minimum light will be there. So to measure that you have to do like OJP, you need to check all the 4 transient states. Yeah.
So I did it 2 times with the same sample.
So how much difference you got?
First time it was 0.617. 617.
Okay.
Second time 0.562.
562, there is much difference. It depends on, because we are doing in light also. And also while doing, you need to shake, a gentle shake in it, because cells will settle down more. So just a gentle shake. But much difference it won't show. If the cells are healthy, it will show the minimum differences. Yeah.
The ideal value is like 0.7 around.
Yes. It's like if you take the spectrophotometer values, how you will take, triplicates you will take, how you will measure, 3 times you will do. So here there you will do blank, right? Here you do auto gate. Yeah.
చాలా మంది వచ్చి ఇచ్చారు కాబట్టి దానికి క్లిప్స్ అయిపోయాయి సార్. దానికి ఒక 20 minutes మళ్ళీ చేయడానికి
20 minutes
బ్యాక్ ఇంక్యుబేషన్ చేయడం చేయొచ్చా మళ్ళీ, లేదా ఈ రోజు అంతే?
చేయొచ్చు. అంటే టైమింగ్ పాయింట్స్ కూడా ఉంటాయి. ఫర్ ఎగ్జాంపుల్ లైట్స్ కూడా ఫ్లక్చుయేటింగ్ లైట్స్ అవి ఇవి ఇస్తారు వాళ్ళు, ట్రీట్మెంట్స్ ఇస్తుంటారు. సో డిఫరెంట్ ఫ్లక్చుయేటింగ్ లైట్ కండిషన్స్లో డిఫరెంట్ టైం పాయింట్స్ వాళ్ళు తీసుకుంటారు. సో డిఫరెంట్ టైం పాయింట్స్ కూడా తీసుకోవచ్చు.
ఇప్పుడు డెమోగా చేయొచ్చు సార్, అంటే
చేయొచ్చు.
చాలామంది
చేయాలన్నా చేయాలి, పర్లేదు. మేబీ ఇప్పుడు ట్రీ బ్రేక్ ఉంటుంది, తర్వాత కూడా చేయొచ్చు.
టీ బ్రేక్ అప్పుడు 15 minutes ఇంక్యుబేషన్ పెట్టిద్దాం, వచ్చిన
కరెక్ట్ ఫాలో అవ్వాలి. ఇది ఏంటంటే డెమో మీకు నేర్చుకోవడానికి. మీరు ఏదైతే నేర్చుకున్నది మీరు మీ ల్యాబ్లో ఇంప్లిమెంట్ చేస్తారు. అప్పుడు ఆ కండిషన్స్ ఫస్ట్ నుంచి ఫాలో అవ్వాలన్నమాట. డే వైజ్ తీసుకోవడం కానీ, లేకపోతే ఆర్ పాయింట్స్ కానీ, సో అలా తీసుకుంటే మీకు డిఫరెన్స్ తెలుస్తుంది. ఇప్పుడు ఏంటంటే ఇది డార్క్లో చేయాలి, లైట్లో చేస్తున్నాం మనం. సో డిఫరెన్సెస్ అయితే ఉంటుంది, అక్యురేట్ ఉండవు వాల్యూస్.
ఇప్పుడు ఈ ఆల్గేని ఏ మీడియం ద్వారా గ్రో చేస్తున్నారు?
ఇక్కడ మేము యూజ్ చేసేది ట్యాప్ మీడియా, ట్రిసెస్టేట్ ఫాస్ఫేట్ మీడియా. Plants usually few days will grow, right? But microalgae it will grow in 3 days, especially this organism.
అది అంటే ఫస్ట్ ప్రోగ్రామ్లో రాఘవేంద్ర సార్ రెడ్ టైడ్ గురించి మాట్లాడారు కదా.
ఏది చెప్పారు?
రెడ్ టైడ్.
ఓకే, నేను లేనప్పుడు బయటకు వెళ్ళి కొంచెం పని ఉండే. ఓకే, డిఫరెంట్ ఆల్గేస్ ఉంటాయి. రెడ్ ఆల్గే అంటే ఫర్ ఎగ్జాంపుల్ డిఫరెంట్ ఆర్గానిజమ్స్ ఉంటాయి ఆల్గేలో. రెడ్ ఆల్గే అంటే ఏంటంటే అది అక్యుములేట్ చేసే పిగ్మెంట్స్ వల్ల ఇట్ అపియర్స్ రెడ్ ఇన్ కలర్. ఉంటాయి, నాకు తెలియదు, మరి నేను వినలేదు. అడుగు, ఏం డౌట్స్ ఉన్నా అడగాలి, పర్లేదు. ఏ చిన్న డౌట్ ఉన్నా అప్పటికప్పుడు అడిగేసి పర్లేదు. నేర్చుకోవడానికే కదా మీరు వచ్చింది.
ఓన్లీ రెడ్ ఒకటే కనిపిస్తుందా? లైక్ 3 లైట్స్ ఉన్నాయి కదా, ఓన్లీ రెడ్ ఒకటే
రెడ్ వస్తుంది, 3 నుంచి రెడ్ వస్తుంది. అంటే ఇది ఏంటంటే ఇప్పుడు ట్యూబ్ ఇట్లా ఉంది, ఒక సైడ్ నుంచి లైట్ వస్తే ఇక్కడ మెజర్మెంట్ ఉండదు. సో 3 సైడ్స్ ఏంటంటే కవర్ అయిపోతుంది, మొత్తం 360 degrees కవర్ అయిపోతుంది. సో వాల్యూస్ అనేవి అక్యురేట్ వస్తాయి.
ప్లాంట్లో మనం అది అంటే సాఫ్ట్ సైడ్ డార్సల్ అప్పర్ అప్పర్ సర్ఫేస్లో క్లిప్ పెట్టాలా, లోవర్ సర్ఫేస్ అప్పర్ లైట్ ఎక్స్పోజ్ అవుతుంది క్లిప్ పెట్టాలి, పైన పెట్టాలి. కానీ కొన్ని చోట్ల వెనక పార్ట్ కూడా లైట్ ఎక్స్పోజ్ అవుతుంది. దానికి మళ్ళీ వేరేగా చేస్తారు. మెయిన్గా అయితే ఫోటోసింథసిస్ మెయిన్గా ఏంటి పైన లైట్
మీకు ఈ ప్లాట్ చేసే దాంట్లో ఏమైనా డౌట్స్ ఉంటే అడగండి. ఎక్స్పోర్ట్ చేసి చూపించాడు చెప్పి.
So this you can deconvolute later. Just what you have completed, right?
Are you completed, right?
Yes, done.
So we can stop the
Have
live.
a break, yeah.
Live we can stop live.
Yes.
Completed the procedure.
Yes. Once confirm with Jyotiba if he wants to.
So told it after
It's
completion
done.
of you.